Good evening, friends. Welcome to the first class of Target Prelims. अभी तक जो भी क्लास आपने मिस करी हो, please don't miss any class from now on. Everything that is needed for preliminary examination, would be covered from now. Again, even if you have missed previous classes of current affairs and all, we would be repeating those topics also. We would be revising those topics also. So please ensure that you don't miss any class from today. Okay. So online students, are you able to hear me? The people who are watching it online, एक बार मुझे confirm कर दीजिए. प्लीज़ ऑनलाइन स्टूडेंट्स एक बार कमेंट में कंफर्म कर दीजिएगा क्या मेरी आवाज़ आ रही है. ओके, तो बिफोर वी स्टार्ट द क्लास, let us spend 10-15 minutes in let me spend 10-15 minutes in explaining what the structure of the program would be, what the structure of Target Prelims program would be, what are the things we would be covering, what will be the timelines, and how many tests, how many RTFQs, all those aspects we would be briefly talking about, we would briefly be discussing. जल्दी सेटल हो जाइए सब लोग, इधर बैठ सकते हैं, इधर ये स्क्रीन है आपके लिए, है ना. आज इतना क्राउड है, मुझे नहीं लगता किसी को सर्दी लगेगी, है ना. ऑलरेडी गर्मी लग रही है थोड़ी सी, चलिए अच्छा है, जल्दी सेटल हो जाइए और कोशिश करिए थोड़ा टाइम पे आएंगे, सो दैट बच्चे ज़्यादा है ना, अगर लेट आओगे तो सेटल होने में दिक्कत होगी. तो लेट मी एक्सप्लेन द प्रोग्राम थोड़ा सा. तो देखो, as you must have seen this, we would be exhaustively covering all current affairs that is relevant for preliminary examination, but for a few topics. For example, for science and technology and environment, we would be covering everything, even the basic static portion. So for environment, biodiversity and climate change, you need not cover anything at all. I will be ensuring that class 12th NCERT के जो आखिरी के 6 chapters हैं, they are there in your notes. We are doing questions on them. I will be ensuring that जो कोई भी basic book है, कोई भी standard book है, उसमें जो भी relevant चीज़ है for environment, it would be there in your notes. Okay, so please understand, book plus current affairs वो थोड़ा सा ज़्यादा होगा, 300-400 pages होंगे, but that is your entire syllabus for environment, biodiversity and climate change. You won't need to do anything at all, be it NCERT, be it any standard book, be it any current affairs. Okay, I'll go through all the current affairs, all the material which is available, and if I find anything relevant which I may have missed, I would add it in your notes. So that is my responsibility. Similarly, when it comes to science and technology, just go through class 9 and class 10 NCERT. पांडे जी गेट खुला रहने दीजिएगा, थोड़ा सफोकेशन वगैरह ना हो. जस्ट गो थ्रू क्लास 9 and class 10 NCERT. Browse through it. All of you have studied that in your school days. तो एक बार just browse कर लोगे, that would be good enough. I'll be covering those sections also, the relevant sections from there also, but not everything. पर फिर भी 9-10th आपको एक बार पूरा अच्छे से देख लेना चाहिए. But when we come to other subjects, for example, economy, Indian polity, इसमें ना what I expect you to do is history, geography, polity, economy के basics आपको खुद करने. I'll not be covering history in the classroom, history and culture in the classroom, बस उसका current affairs cover करेंगे. I'll not be covering geography in the classroom, बस उसका current affairs cover करेंगे. I'll not be covering Lakshmikant in the classroom, only current affairs we would be doing, and I'll not be covering basics of economy in this classroom. We'll be doing current affairs of that. Okay, but we would be revising basics through questions. Before every class, जैसे हम जब economy की class में आएंगे, and let's say we are discussing on that day, we are talking about economic growth wala topic. So we'll talk about what are all the things which have happened recently in economic growth. But before that, I'll make you solve some questions, 15-20 questions, so that you are able to judge whether you are comfortable with basic portion or not. Fine. So all current affairs, but for science and environment, even the basic thing would be done here. ये बात समझ में आई? First thing. So what do you have to study other, if you are preparing for preliminary examination, what do you have to study other than this history, geography, polity, economy? ये basics, इनके भी बस basics, no current affairs material, nothing. Just I'll not be able to teach this in the classroom कि what is the difference between GDP and GNP. है ना, ये बेसिक्स आपको आने चाहिए। अगर इसमें आप अनकंफर्टेबल हैं तो देयर इज अनदर प्रोग्राम फॉर प्रिलिम्स, वो जो बेसिक्स पे फोकस करता है, प्रिलिम्स मास्टर्स प्रोग्राम। योर यू शुड नॉट बी जॉइनिंग दिस प्रोग्राम, यू शुड बी जॉइनिंग दैट प्रोग्राम। फाइन, ये तो दिस इज द फर्स्ट एस्पेक्ट, व्हाट वुड बी डन। इसमें कोई डाउट है किसी को? नहीं, सब लोग कंफर्टेबल है। नाउ कमिंग टू, we would focus on solving lots of MCQs. In every class, I assure you, we'll be solving around 25 MCQs. Then when a topic is done, let's say I'm planning to finish science and technology by 22nd, 23rd January. After that, we'll have a full test on science and technology of 100 questions, entire syllabus. And after that, we will also be solving 500 questions on science and technology. Revised through 500 question series में we would be dealing with that. So our focus would be a lot on solving MCQs, and there, other than revising the basic portion, the current affairs portion, we would also learn various tricks that we use to eliminate options. I personally do not rely a lot on that. What I want is आप लोग इतने अच्छे से prepared हो कि tricks की जरूरत ना पड़े, but फिर भी no one can know 100 out of 100 questions in UPSC. So tricks are useful. Through those questions we would be learning those tricks as well. ये बात समझ में आई? Then by April 30 we'll complete our entire syllabus, we'll complete our MCQs and the sectional test. In the month of May we'll have 4 full tests. तो बात समझ में आई? After every section, in every class we'll be doing 25 questions. After every section we'll be having a full test. Then for these 3 subjects, science, environment, economy, I promise you that we will be doing 500 questions, revised through 500 question series for these 3 topics. Pakka. I'm not promising it for other subjects, but for these 3 things we'll be doing RTFQ. For these we'll continue doing test, and वो 25 questions in every class we would continue doing that. इसमें मैं RTFQ promise नहीं कर रहा हूँ, मैं कोशिश करूंगा, but I'm not promising it as part of this program. Okay, then anything else, any doubt related to the program? कोई कंसर्न, कोई इशू नहीं? सब लोग ऑनलाइन वाले बच्चों को कोई डाउट है? क्या ये क्लासेस हिंदी मीडियम में भी अवेलेबल हैं? नहीं, ये क्लासेस हिंदी मीडियम में अवेलेबल नहीं है, एंड आई विल नॉट बी एबल टू कमिट टू दैट. सर, आर यू लॉन्चिंग एनी प्रीलिम्स स्पेसिफिक क्रैश कोर्स बाय द एंड ऑफ दिस मंथ? देखो, प्रीलिम्स यही है, दिस इज प्रीलिम्स स्पेसिफिक क्लास क्रैश कोर्स विच वी आर स्टार्टिंग. यू कैन ज्वाइन इट एनी टाइम यू वांट. The portion that you miss, you'll be able to get videos for that, you'll be able to watch it online. Now coming to number of classes, how many classes we'll have? So for the first two, officially we are going to have 3 classes every week, Friday, Saturday, Sunday. But for the first 2 weeks, थोड़ा classroom का constraint है, so I'm not, I'll only be able to take 2 classes. So this 7th and 8th, and then 14th and 15th पे, 15th को we are only having 2 classes a week. After that we will have at least, I'm using the word at least, please note, 3 classes every week, Friday, Saturday, Sunday, जो कि 5 to 9:30 चलेगा, जिसमें 5 to 7 half an hour का break, and then 7:30 to 9:30 we'll have 2 sessions. Okay, so this is about number of classes. Now sir, क्या ये क्लास ऐप पे अपलोड होगी? हाँ, जो people who are watching it online, you'll be able to do it online, you'll be able to do it on the app or through the website. मैपिंग एंड गवर्नमेंट स्कीम्स, यस डेफिनेटली गवर्नमेंट स्कीम्स, हम सारी गवर्नमेंट स्कीम्स जो रिलेवेंट हैं करेंगे, सेक्शंस में करेंगे. साइंस एंड टेक्नोलॉजी जब डील कर रहे हैं तो साइंस वाले करेंगे, एनवायरनमेंट जब डील कर रहे हैं तो एनवायरनमेंट वाले करेंगे, इकोनॉमी जब डील कर रहे हैं तो इकोनॉमी वाले करेंगे. अदर देन दैट मिसलेनियस थिंग्स, फॉर एग्जांपल विमेन इश्यूज, चिल्ड्रन इश्यूज, इन पे भी बहुत सारे स्कीम्स, प्रोग्राम, इनिशिएटिव्स रहते हैं, वो भी हम सोशल जस्टिस वाले एस्पेक्ट में डेफिनेटली करेंगे. सर, पीएमपी दोबारा शुरू होगा? हाँ बेटा, प्रीलिम्स मास्टर्स प्रोग्राम वुड स्टार्ट अगेन इन February. ओके, नाउ will we be getting test plan? Test plan after we finish. देखो, questions in every class, and after we finish a portion, right after that you'll have that portion का full test. That is the test plan. I have not kept it strictly weekly. जैसे Pre-Cum-Mains वाले बच्चों का weekly test चल रहा है, but for target prelims, people who have joined only target prelims program, I have not kept it strictly weekly test. I have kept it हर क्लास में क्वेश्चंस करेंगे, और जब वो पोर्शन फिनिश हो जाएगा, we'll do a full test after that. Okay. स्टैटिक का क्रैश कोर्स, हाँ स्टैटिक का क्रैश कोर्स, पीएमपी प्रीलिम्स मास्टर्स प्रोग्राम, दैट इज गोइंग ऑन ऑलरेडी. और उसे दोबारा फरवरी में भी स्टार्ट करेंगे. Stick with English, sir. Please stick with English. Yes. This is supposed to be an English class. I'll stick to English only. Okay. Any doubts? Any concerns? Any issues? नहीं, शुरू करें. Should we start? What will be the sequence of the subject that we would be going through? So sequence would be this. First, we will do science and technology. Then we will start with economy. So by 25, I plan to by 25th January, I want to finish science and technology, so that from 26th to 31st, you people can focus on revising basics and everything. I want to start from 1st of February. I want to ensure that Economic Survey of India chapter wise is done, along with other current affairs related to those topics. So in the first 10 days of February, we will be doing Economic Survey of India and budget, along with other current affairs related to economy. So I'm expecting that by February 15, in worst case, we would be done with economy, as well as economy ka revised through 500 questions on economy will be done with. Then we'll go to environment, biodiversity and climate change. This we should be finishing it by March 10. Then from March 10 to April 10, around 1 month, in around 1 month, we would be talking about polity and international bodies. Please note that here we will not be doing international relation. International relation pre में questions नहीं आते. Pre में questions आते हैं international bodies से. United Nation pre में question आते हैं, IMF pre में question आते हैं, WTO से pre में questions आएंगे, various groupings, the reports that we will be doing here. So this will go on till around March 10. उसके बाद जो miscellaneous चीजें हैं, miscellaneous like we would have things like, let's say, mapping, we would have culture, we would have some other small portions, we would be dealing with all those aspects. Fine, जो भी miscellaneous छोटी छोटी चीजें बचेंगी, we would keep doing that, plus any updates. As I've told you, आपको current affairs नहीं touch करना है कहीं और से. It's my responsibility. You just ensure that every day 45 minutes to 1 hour you're going through the newspaper. Do not skip newspapers. But any other material is not your responsibility. That is my responsibility. My material would be more than what is needed. Your complaint at the end would be, if you are not revising, कि सर बहुत सारा पढ़ाया है. Your complaint would not be कम पढ़ाया है. Fine. And वो complaint लेके मत आना कि बहुत सारा पढ़ाया. बहुत सारा पढ़ाएंगे हम, सबसे ज़्यादा पढ़ाएंगे हम, सबसे ज़्यादा क्वेश्चंस करेंगे हम, सबसे ज़्यादा सीखेंगे हम, सबसे ज़्यादा प्रिपेयर्ड हम रहेंगे. If you want that I should be finishing entire science and technology in 20 pages, I won't be able to do that, because that's a scam. You cannot study science and technology like that unless you understand the basic concepts. If you want that those like 50-20 pages में मैं पूरा current affairs आपके लिए cover करके दूं, I won't be able to do that. For people who are expecting something like that, please join somewhere else. I would ensure that you are best prepared for the examination. And उसके लिए जितना पढ़ाना पड़ेगा, जैसे पढ़ाना पड़ेगा, वो होगा. Similarly, I've said we'll have at least 3 classes every week, Friday, Saturday, Sunday. If I feel like I'm missing these deadlines, I'll start another class on Thursday. Please note it. मैं ये नहीं बोल रहा कि मैक्सिमम 3 क्लासेस होंगी, मैं बोल रहा हूँ मिनिमम देयर वुड बी 3 क्लासेस. मैक्सिमम पे लिमिट नहीं लगा रहा हूँ. बट व्हाट आई एश्योर यू, आप लास्ट ईयर मैंने प्रीलिम्स के बाद वीडियो रिलीज किया था. फ्रॉम दिस प्रोग्राम देयर वर अराउंड 44 क्वेश्चंस व्हिच वर आस्क्ड इन द एग्जामिनेशन. इफ ऑफ दोस 44 क्वेश्चंस आप 35 भी सही कर सकते हो. 44 questions from these notes from the section that we are starting today were asked in the examination. That is there. I think description में भी वो video होगा where this is there कि हमारे class notes से या हमारे program में से हमने screenshot ले लेके एक video बनाया था. You watch those videos. So by giving you 3 days, 50% of your preparation would be done. Rest of the preparation रहेगा history, geography, polity, economy का basics करना, वो आपको करना पड़ेगा. Okay, basically there are some beautiful lines by Harivansh Rai Bachchan ji. My class somehow fits on those lines. He says कि जगती की शीतल हाला सी पथिक नहीं मेरी हाला, जगती के ठंडे प्याले सा पथिक नहीं मेरा प्याला, ज्वाल सुरा जलते प्याले में, दग्ध हृदय की कविता है, जलने से भयभीत न जो हो, आए मेरी मधुशाला। तो मेरी क्लास में सिर्फ वो लोग आए जो पढ़ने से नहीं डरते हो, जो चाहते हैं ना कि बस कोई ट्रिक ट्रिक बता के हमारा प्री क्लियर कर दो, वैसा नहीं होता। जो चाहते हैं ना कि 10 सबसे इंपॉर्टेंट टॉपिक्स करा के हमारा प्री क्लियर कर दो, यूपीएससी को अनप्रेडिक्टेबल पब्लिक सर्विस कमीशन इसलिए बोलते हैं। दोस टेक्निक्स, दोस ट्रिक्स, दोस फालतू थिंग्स डजंट वर्क। यू हैव टू बी फुली प्रिपेयर्ड। तो पीपल हु आर नॉट रेडी टू वर्क हार्ड, वो मेरी मधुशाला ना आए। People who are really sincerely ready to work hard, please come and attend these classes. Please do a test on a timely manner. Please be prepared for RTFQ. जैसे हम साइंस का अराउंड 22nd 23rd जनवरी को हम RTFQ करना शुरू करेंगे, 500 क्वेश्चंस करना शुरू करेंगे। बाय दैट टाइम यू शुड हैव फिनिश्ड एंटायर साइंस एंड टेक्नोलॉजी, व्हाट NCERTs and the classes. Your entire preparation of science and technology would be that. Other than that, in April we may give you some updates जो अभी जनवरी फरवरी मार्च में न्यूज में रहेंगी चीजें, वो तो अभी नहीं कवर हो जाएगा जो फरवरी मार्च अप्रैल में रहेगा। अदर देन दैट यू विल ओनली हैव टू डू दैट। ओके, तो जलने से भयभीत ना जो हो, वही आईएएस क्लास। फाइन, तो शुड वी स्टार्ट इट? एनी अदर डाउट्स गाइस? एनी अदर डाउट्स, एनी अदर इश्यूज? नो, ओके, let me start with today's class. Here we have briefly mentioned what are the sources that we are covering. So Express Hindu, Down to Earth, योजना, कुरुक्षेत्र, PIB, these are the primary sources we cover. मैं Pre-Cum-Mains में we also deal with live law and ORF, but live law and ORF issues are mostly main centric, so we are not covering those things. So other than these six main sources, Economic Survey of India and budget, as and when they come, we would be dealing with them. Okay, so let us start with our first class, that is on biotechnology. गलती से मैंने यहाँ 25 से ज़्यादा क्वेश्चंस दे दिए हैं. I wanted to limit to only 25 questions in every class. Today we have around 40 questions. चलो थोड़ा ज़्यादा कर लेंगे. But all those questions are relevant, are based on what UPSC has asked in the past. The depth to which I will go in these classes would also be based on the depth of questions where UPSC has gone. Like biotechnology में हम PhD कर सकते हैं. Forget biotechnology, हम किसी biotechnology के छोटे से पोर्शन में भी PhD कर सकते हैं. But that is not what we'll do. To what depth we go, it is based on what has UPSC asked in the past. उससे एक step आगे, so that next time if they go slightly in more details, we should be able to deal with that. Okay, should we start? Biotechnology, the first topic. और सब अच्छे से पढ़ेंगे. अभी the questions which we have given, I'm going to ask you those questions. They are from your class, they are from your notes, they are from what we are going to discuss. और वहां गलती होगी तो मुर्गा बना दूंगा मैं. Okay, now we will stick to English, nothing in Hindi. Else some people are going to kill me. Biotechnology. What is meant by biotechnology? In very basic terms, biotechnology means using biological resources, systems, etc. to produce something that is useful for us. For example, all of us consume curd, dahi, right? How do we prepare curd? We take some bacteria from the previous curd, we add it in the milk. Those bacteria work on the milk and produce curd. They multiply and they also process the milk to produce curd. Now, curd is a useful product for us. How was it produced? It was produced it was produced using some living organisms, using some biological resources. So this is an example of biotechnology. So biotechnology is not something new. We have always been using it. Think of alcohol. How do we make alcohol? If we have sugar, we ferment it using some yeast. So here also we use living organism to produce something which is useful for us. So making of alcohol is also an example of biotechnology. So in very basic terms, biotechnology is the use of biological processes, organisms or systems to manufacture products intended to improve the quality of life for humans. We have seen an example of how very basic things that we have been using since time immemorial, be it alcohol or be it curd, is an example of biotechnology. In this class, when we'll talk about modern biotechnology, we'll see how advancement in biotechnology have allowed us to modify the existing living organisms to make them more useful for us. For example, हमने cotton को modify किया BT cotton, so that it is in itself able to fight some pest and the production is higher. So we have modified some living organisms to make them more useful for us. That is also an example of biotechnology, but that is modern biotechnology. The biotechnology can basically be classified into two broad types: the classical or traditional biotechnology or the modern one. Classical or traditional biotechnology का हमने example देखा, क्या देखा? Where we were using curd, where we were using yeast to make alcohol. Now notice something: when we used curd or when we used yeast, here we did not, bacteria, curd में हमने bacteria use किया, और when we used yeast we did not modify these organisms, we use these organisms as it is. But suppose tomorrow we do this, tomorrow we change this bacteria so that rather than taking one whole night, it is able to produce curd within 1 hour. It is able to produce curd within 1 hour. Then this would not remain an example of classical biotechnology. This would become an example of modern biotechnology, where we change the system, we change the living organisms to make the system more effective for us, more beneficial for us. So biotechnology can be classified into two broad types. One is the classical biotechnology, where we are using this. Other is the modern biotechnology, where we modify the living organism, be it through genetic modification or hybridization, to make them more useful. Now let's take this example of penicillin. How penicillin is produced? So scientists realize that there's a mold, penicillium mold. It produces this antibiotic penicillin. So now what they did, they produced this mold in large quantities in a container where sugar was there, and this mold multiplied there. Then from this mold they extracted penicillin. And penicillin, we all use penicillin as antibiotic. Is this an example of modern biotechnology or classical? Classical. We have not modified the mold at all. We have not modified the mold at all. We have produced it in large quantity and have extracted what from it? We have extracted the penicillin from it. So this is also an example of classical biotechnology. So please understand कि ऐसे differentiate नहीं करना है. Okay, modern medicine should be an example of modern biotechnology. No, that's not the case. It is how we are using the technology, like whether we are modifying the system or not, that would determine whether it is biotech or not. Fine, ये बात समझ में आई. Now here we have examples of genetic engineering in modern biotechnology. So we will talk about BT cotton, BT brinjal, or golden rice. All these are example of how modern biotechnology has been able to used to produce crops which are more useful for us, which are more beneficial for us. Here we'll talk about other genetically modified living organisms. Here we'll talk about gene therapy, how diseases which were not treatable earlier have been treated using genetic modification. That will also be an example of modern biotechnology. Now please note that modern biotechnology is not just genetic modification. अभी तक तो हमें यही परसीव हो रहा था कि till now we were thinking कि modern biotechnology is the genetic modification वाला. It's not just that. Modern biotechnology may also include things like polymerase chain reaction, PCR. What do we do in polymerase chain reaction? आपने RT-PCR test कराया है? Today we'll understand it properly. उसका one of its component is polymerase chain reaction, in which we get a piece of DNA and we make lots of copies of it. We make a lot of copies of those polymerase chain reaction. That is also an example of modern biotechnology, but for that to happen, we could not do it traditionally. For that to happen, we needed a system of tissue or cell culture which has been developed only in modern times, only in recent times with more advancement, and thus this would also be an example of modern biotechnology. Fine. Now we want to understand the basis of biotechnology. Now for next 15-20 minutes, whatever we study will basically be the core of biotechnology. If you understand this, you will understand each and every topic which we are going to study in this class. Each and every topic would just become very factual for you. You'll just see कि अच्छा ये हो रहा है, ये हो रहा है, ये हो रहा है, because you would have understood the basic concept, how this modern biotechnology is functioning. Okay, so let's understand that. To understand this modern biotechnology, first let's understand why am I like this? Why do I have hairs of some color, or skin of some color, or eyes of some color? Why does my body produce some protein, or why is my behavior of this, of what I am right now? All my characteristics are defined by the genes that I have taken from my parents. All my characteristics are defined by the genes that I have taken from my parents. So basically, when an egg is fertilized, it has chromosomes from both mother and father. Those chromosomes bring the genetic material to the progeny. So what we get from our parents are these chromosomes. Now on these chromosomes is there something called DNA, deoxyribonucleic acid. On these chromosomes is the DNA. DNA is known by its double helix structure. You must have seen this kind of structure. Abhi we will go into the details of this structure, but DNA is known by its double helix structure, which is, this DNA is made up of nucleotide. And what is nucleotide? Nucleotide इसका एक इस DNA is basically a polymer, where it's a polymer of nucleotides, and this nucleotide is basically a nitrogen base, a sugar, and a phosphate group. Okay, so if you will look at this DNA would basically be a nitrogen base. अब ना let me open this up. ये double helix structure है. Let me open this DNA and let me just make it a staircase like this. This is DNA. This is there in your notes. So focus on understanding. This is DNA. मैंने इसे जो double helix structure में था मैंने इसे खोल दिया. Let's keep it like this. So I'm saying that this is a polymer of nucleotides. And what are nucleotides? Nucleotides basically are nitrogen bases. तो यहाँ पे एक nitrogen base है. It is attached to a sugar. It is attached to a phosphate group. So what you will have is a series of nitrogen base, sugar, phosphate group. Nitrogen base, sugar, phosphate group. So what you see that we have a repetition of nitrogen base, sugar, and phosphate group. Polymer of this. On this side also, there is a base, sugar, phosphate group. Base, sugar, phosphate group. So another set of nucleotides. Base, sugar, phosphate group. DNA is a polymer of nucleotides. And what is nucleotide? Nucleotide are these things combined together. Nitrogen base, sugar, यहाँ पे deoxyribose sugar है, that's why it's deoxyribonucleic acid, DNA, and a pentose group. Oh, sorry, phosphate group. If you'll go to this image, you'll understand this. जो ATCG है, these are nitrogen bases. So G is a nitrogen base connected to a sugar, which is connected to a phosphate group. This is a nucleotide. This is a nucleotide. This is a nucleotide. A series of nucleotide, a polymer of nucleotide is deoxyribonucleic acid. Okay, अभी इस पे और आते हैं, but let's go to the basics. It's made up of this. Now, in our DNA, there are billions of these nucleotides, billions of them. Okay, a subsection of it, a subsection of it. जैसे ये पूरा डीएनए है हमारा, a subsection of it is called gene, which if it encodes for a protein. So why is my hair, why are my hairs of a particular color? Because my body produces a particular protein which is giving color to my hair. Where is the information related to that protein in my body? It is there in my DNA. On my DNA there are genes. Gene 1 may be this, gene 2 may be this, gene 3, gene 4. In the same way, in this way we have around 20,000 genes in our body. And these genes are basically the information about what protein would be formed in your body. So जो fundamental unit of hereditary है ना, it is this gene. Fundamental unit of hereditary, if I am getting a feature from my mother or from my father, that feature would be in the form of these genes. On the DNA, there can be multiple genes. On the DNA, there can be multiple genes. Okay, अभी तक बात समझ में आई? Now, we have chromosomes that we get from our parents, right? अगर आपकी लाइफ को देखा, if I look at you, like let's say I am looking at Winkin is an entire organism. She is made up of various organs. She is made up of various organs. Heart है, lung है, skin है. These are all organs. Those organs are made up of tissues. Those tissues are made up of cell. So a cell is considered fundamental unit of life. ये हम अभी basics of biology थोड़ा पढ़ेंगे, उसमें we will explain you this in more detail. A cell is fundamental unit of life because it is capable of doing everything that is needed for survival. एक सेल जिंदा हो सकता है, इसका सब यूनिट we cannot say that it is alive. Why? Because it won't be able to do everything that is needed for survival, for living. Right? Inside every cell that we have, there are few exceptions as well. We have a nucleus which has this chromosome, which have these chromosomes that we get from our parents. Now, this is, did you understand your genes wala aspect? Did you understand your DNA wala aspect? Now please note this, the basic reason or the core of biotechnology is this. In all living organisms, be it a human being, be it a mosquito, be it a rat, be it a bacteria, a small bacteria which you are not able to see, their hereditary information is stored in this way only. If you take a bacteria and go into its nucleus and take out its DNA, it will have this kind of structure. And it will have genes like this producing some protein. The protein may be different, but the way the protein is stored is exactly, the way the information about the protein is stored is exactly in the same manner. Exactly in this manner. Okay, so what is the idea? Idea is this, ki bhai, if in a bacteria also, in a plant also, in a mosquito also, if in a dog also, in a buffalo also, we have the same way in which genetic information is stored. Same structure. Can we not take some feature from there and insert it here? It's possible because the way information is stored in these living organisms is in the form of DNA. They would also have these nucleotides जहाँ पे information store होगा. See, how is information stored in our body? In this form, in the form of these molecules. Suppose there was some other living organism where information was stored in some other way altogether. Let's say इस form में नहीं होता. There the way of storing information is in the form of some other chemicals, some other structure, इस तरह का कोई structure है, and some other chemicals are there. Then taking a feature from here and inserting it here would not have been possible. But I'm saying कि all living organisms except some RNA viruses, some viruses which use RNA to store information. There are some viruses which are DNA viruses. वहाँ पे भी but except some RNA viruses. जैसे is COVID-19 virus an RNA virus? Yes. So information, COVID-19 virus produces various protein. हम कल पढ़ेंगे. Tomorrow we will be dealing with health. In health, we will talk about various diseases and we will talk about the structure of COVID-19 virus and all. But वहाँ पे they are not storing information, they are not storing genetic information in this manner. But in all other living organisms, except RNA viruses, in all other living organisms, information is stored like this. This is the core of biotechnology. Now understand this. I want some feature from some other living organism. I can take some gene from there and insert it in a human gene. Those features would be able to come to this human. I always give this example of golden langur. You know what golden langur is? You know what golden? उसके golden furs होते हैं. It's found in Northeast India. It is endangered. उसको भी अभी environment biodiversity में we would study that. Now suppose I wanted, or my parents wanted कि मेरे golden hair हो. Naturally. है ना? ऐसे नहीं कि बस वो golden color कर लिया. They wanted कि I should have golden hair. I should have had golden hair. What they could have done is, when life starts as a single cell, I also started as a single cell. When my mom's eggs were fertilized by my father's sperm, that was a single cell. It has a nucleus. It had a DNA, which was which took some features from my father, some features from my mother. If they had removed gene which is giving me this color and could have taken, अब उसका सोचो, गोल्डन लंगूर का डीएनए कैसा होगा, उसका स्ट्रक्चर भी ऐसा था, वहां से they took the gene and inserted it here. तो अभी I could have had golden hair. Why was it possible? It was possible because the way information is stored, the way genetic information is stored, is this in all living organisms except some RNA viruses. The way information is stored is like this in all living organisms except some RNA viruses. Right? Let's say I wanted कि भाई golden hair नहीं, ऐसा होना, my saliva should be venomous. King cobra है, मैं जिसको भी काटूं थोड़ा saliva छोड़ दूं, king cobra वाला सीन हो. Suppose I wanted that. Was that possible? Snake is a living organism. It has DNA. It has DNA. So when it is producing venom, so for that venom also there must be some genetic information by which it is producing that venom. So if I was given that gene, then अभी कोई मुझे परेशान करता, मैं बस काट लेता. राइट? काटता तो थोड़ा सलाइवा तो जाता ही, उसकी लग जाती. So understand this. Now think of BT cotton. Cotton was a plant. It was being attacked by various pests. It was being attacked by various pests, because of which what was happening? The production was going down. Scientists found that there's a bacteria, soil bacteria, which produces a toxin which can kill those pests. There's a soil bacteria which produces a toxin which can kill those pests. Now, scientists said, उसका जीन होगा ना, let's take out that gene and put it in cotton. तो जो कॉटन का डीएनए था, it was modified and a gene, let's say ये अपने बैक्टीरिया का डीएनए है, इसमें this was the gene which was responsible for production of that toxin. इसे बोलते हैं हम cry1AC gene. We'll talk about that. We take this gene and insert it here. Now cotton would start doing what? Cotton plant itself would start producing that toxin. This became BT cotton. This became BT cotton. How did this? What happened here? ये बैक्टीरिया जो था, that was BT bacteria, Bacillus thuringiensis bacteria. उसका एक फीचर लेके हमने कॉटन में डाल दिया. नाउ माय कॉटन प्लांट itself is producing that toxin, and now when the pest come, they are not able to come, they die, they're not able to attack this cotton plant. Why is this possible? This is possible because genetic material is stored in the same manner. Again, in bacteria, if the rather than in the form of nucleotides, the material was if the genetic information was stored in some other way altogether, then taking it and inserting it here would not have functioned. Why would it have not functioned? It would not have functioned because the way our body is producing protein is different. We would not have been able to produce that protein, but because the entire system is same, we are able to do it. So think of a mosquito, वो छोटा सा मच्छर जो आप ऐसे मार देते हो. It also has DNA. It also has DNA, which has these base pairs, ATCG base pairs, which has this sugar, deoxyribose sugar, which has this phosphate group, and it is also producing protein. So tomorrow, if you want that, you should be able to produce some protein which this mosquito is able to produce. Genetic modification may be possible. Got it? Clarity? Now there are some doubts. Are the number of base pairs? हाँ, अभी we'll talk about number of base pairs कितने हैं, सारी चीजें कितनी हैं. We will go to that. But first thing is you understand कि ये है क्या? How is modern? What is the basis of modern biotechnology? What is the core of modern biotechnology? I told you, no, spend this half an hour in understanding these basic concepts, and you will be able to understand everything. अब ना understanding BT cotton will not be a challenge. क्या किया हमने? बैक्टीरिया से जीन निकाला, कॉटन में डाला. Now cotton is producing what bacteria was producing. Bacteria was producing a toxin. Cotton will be producing a toxin. Suppose tomorrow you want कि your body should be producing penicillin at its own. Your body should be producing penicillin at its own. Naturally antibiotic पैदा हो. Mold था वो. It has some gene which is leading to production of the penicillin. You take it, insert it in your body. Right, this is the core of it. तो इसे ना थोड़ा सा systematically, technically पढ़ लेते हैं. अभी we did it in a very informal structure. Let's go to basic understanding of genetic material. So first is gene. As we said, कि inside a cell there is a nucleus. Inside the cell is the fundamental unit of life. There's a nucleus. In the nucleus, you see inside the nucleus we see these chromosomes. We have this chromosome. On the chromosome, we have DNA, and a sub part of this DNA is called gene. Gene is the fundamental unit of hereditary. So it's the basic physical and functional unit of hereditary. It contains code for a molecule that has a function. A gene ka sub part cannot contain a code for anything. So the smallest unit that can store information for a product is the fundamental unit. Fundamental unit of hereditary is gene. Now genes are located on DNA. It's a short section of genes are short section of DNA that you understand. DNA can be cut and separated, forming a sort of barcode. In humans, gene vary in size from a few hundred DNA bases to more than 2 million bases. Note it carefully. Understand this. What we are trying to say here is, ये अपना DNA था. These are the bases, nitrogen bases. Now, one gene can be formed by a few hundred bases, or it may have millions of bases. So, for more complex protein, the information that would be required to be stored is also higher. We'll have more complex system. For a simpler protein, only a few hundred base pairs would also be enough to store code for that protein. Abhi we'll understand how proteins are formed in our body. We will understand that too. Fine. Now, so as we said, in a base pair, in a human DNA, we can have a around 100 bases together forming a protein or forming code for a protein, or for complex protein, the number of bases that may be required can be up in millions also. Now, Human Genome Project have identified that there are around 20,000 to 25,000 genes in our body. Again, to help you understand this, this is DNA. In our DNA, we have these base pairs, and then we have sugar and phosphate. So, how many base pairs do we have? Around 3 billion. 3 into 10 raised to 9. 3 billion base pairs we have. But एक base pair तो एक gene नहीं है. Multiple genes coming together, multiple base pairs coming together form a gene. Some gene can be formed of 100 base pairs. Some gene can be formed of millions of base pairs. So how many genes do we have? Around 20 to 25,000 genes is something that we have. That is, we have information about 20 to 25,000 proteins which can be formed. एक gene is responsible for one particular product. So we have 20 to 25,000 genes in human beings. जो basic living organisms होंगे, they may have lesser number of genes, lesser number of base pairs. More complex organisms will have more number of base pairs. ये बात समझ में आई? Now, एक छोटी सी चीज और समझते हैं कि सर you said कि this is how the information is stored in all living organisms. How? Nitrogen bases, base 1, base 2, base 3, base 4, again base 1, base 2, base 3, base 4. This is the horizontal part of the ladder, and then sugar, phosphate, sugar, base 1, base 2. This is the vertical part of the ladder. This is the vertical part of the ladder. This is what we had understood. These were nitrogen bases which were bonding together using hydrogen bonds, and here we have sugar and phosphate group. These nitrogen bases are of two types. What are the two types? नहीं, ATCG तो 4 type हो गए ना, प्यूरीन एंड pyrimidine, राइट? आप यहां पे आएंगे, these bases are of two types, purines and pyrimidines. Now purines are made up, जो A और G हैं, they are purines, and जो C and T हैं, जो हम words use कर रहे हैं, C and T, cytosine and thymine, they are pyrimidines, है ना? In DNA we have A, G, C and T, ये 4 ही nitrogen bases होते हैं. In RNA we have uracil. We don't have thymine, we have uracil. So in RNA we have adenine, guanine, cytosine and uracil. But when the pairing takes place, A pairs with T and C pairs with G. In RNA, T तो नहीं रहेगा, तो A will pair with U. So let's come back to this structure. You see this? C G के साथ pair है, A T के साथ paired है, right? This is how this ATCG base pair, A and T are two bases, their pairs. These are called base pairs. This base pair mechanism is used in all living organisms for storing information. Okay, अब देखो, now to understand how protein would be encoded, understand वो computer science. In by using only 0 and 1, we are able to store everything. Suppose you wanted to store 0 and it's a 4-bit system, so you would say this is the code for 0. This sequence is representing 0. Suppose you wanted to store 1, this sequence is representing 1. Suppose you wanted to store 2, this is 2. In binary, this is 2. You wanted to store 3, this is 3. So basically 0 1 0 1 का ऑर्डर आपको बता दे रहा है, अलग अलग एनकोड कर दे रहा है नंबर्स को. इन द सेम वे ATCG pairs will encode for different proteins. तो सारे प्रोटीन में इंफॉर्मेशन कैसे स्टोर्ड है? ATCG के फॉर्म में ही है, बट ऑर्डर, सीक्वेंस, number of pairs, all those things will define a protein in different way. Okay, so information is stored in the form of ATCG in all living organisms, but they may be containing different information. The protein that I may be producing may be different from the protein which a bacteria is producing. Some of the proteins may be same, like or some of the products may be same. हम शुगर को कंट्रोल करने के लिए क्या यूज होता है? इंसुलिन. But that is used in other mammals. That is used. That is needed by other mammals also. So other mammals, a rat also produces insulin. Cow also produces insulin. We also produce insulin. Right. So some of the products that some of the codes may be same. Some of the codes may be different. Fine. Now, so let's come back to हमें ये चीज समझ में आ गई. जीन क्या होता है समझ में आया. Let's understand जीन मैपिंग को थोड़ा सा समझ लेते हैं. What is gene mapping? Gene mapping is basically this on your entire DNA. आपके DNA कितना था? जैसे human beings have 3 billion base pairs. Mapping, okay, this is gene 1, this is gene 2, this is gene 3, this is gene 4. This gene 1 produces this product, is responsible for coding this product. This gene 2 is responsible for coding this product. This gene 3 is responsible for coding this product. This is gene mapping. You identified the genes and then वो gene sequencing में पहले gene sequencing is you sequence the gene. So these are the genes which are there in your body and then mapping them with their products which they are coding is gene mapping. Did you get it? देखो इस साल preliminary examination में आया था DNA fingerprint नहीं DNA barcoding पे सवाल आया था. We'll come to that. That was DNA barcoding. अभी हम जब DNA पढ़ेंगे हम we will talk about barcoding. We'll talk about fingerprinting. But did you understand what is gene mapping? Now DNA. Do you understand this structure? ATCG pairs. Do you also understand this? ATC, GC, AT, CG, AT, CG. You see that C और G ही bond बना रहे हैं, A और T ही bond बना रहे हैं, and CG के बीच में 3 हाइड्रोजन बॉन्ड है. Will not go in the chemistry of it. Just इतना ध्यान रखो कि CG के बीच में 3 हाइड्रोजन बॉन्ड होते हैं, AT के बीच में 2 होते हैं. In case they ask you, वैसे they will not ask you, but in case they ask you, because this is something that's written in NCERT, so we must be aware of it. So this is our structure. So human DNA has 3 into 10 raised to 9 base pair, that is more than 3 billion base pairs. Structure of polynucleotide हमने समझ लिया है. Double helix and ये सारी बातें हमने already देख ली हैं. Now let's come to DNA fingerprinting. The idea of DNA fingerprinting. What is DNA fingerprinting? There are before I go to DNA fingerprinting, there are some doubts online. Let me take care of them. Any specific reason why archaeologists mostly used mitochondrial DNA to identify species from the past? DNA barcoding, जो अभी हम something that we are going to study, are used for identifying species. There we'll see that not only mitochondrial DNA. आप जैसे श्रीचरण Arumugam, you're saying कि only mitochondrial DNA is used. We'll see for different organisms, different portion is being used for barcoding. So we'll come to that. We'll come to that. Anjali is saying, sir, how to know which gene is performing which function? Beta, again, there are many different ways, but let's use a common sense. Suppose I have 25,000 gene in my body. Suppress one gene and see what protein stopped producing in my body. You will know which gene is responsible for which protein. This is one basic way, but scientists use many different ways also, but it's easy to know. Please give examples in English only. Okay. Sir, do human blood cells, Neeraj, no, human blood cells don't have nucleus, or RBCs don't have nucleus. RBCs don't have nucleus, so they may not have these things. Yes, it may have. हाँ, RBC बोला ना, RBC does not. सर किस वक्त पर रोजाना क्लासेस चलेंगी? Classes would be on Friday, Saturday, Sunday, 5 to 9:30. We'll take class from 5 to 7. We'll give a small break and then 7:30 to 9:30 in this way. Okay. So till last year, there are 2 complaints left अभी जो मेरे against है. One, थोड़ा late तक चीजें चलती है. I'll ensure you this time April 30 को your course is getting over. सेकंड कंप्लेन इज लंबी क्लासेस होती हैं, लंबी तो अभी भी होंगी बट आई विल गिव ब्रेक इन अ टाइमली मैनर, तो विल हैव 2 hours क्लास देन अ ब्रेक एंड देन 2 hours क्लास, ओके, सो दैट वो प्रॉब्लम ना रहे। कोई इशू नहीं। ओके, नाउ लेट्स गो टू fingerprinting. Do you understand fingerprint? Your fingerprint is unique for you, that is used to identify an individual. Like my fingerprint would be unique, her fingerprint would be unique, her fingerprint would be unique. In the same way, when we see, we all have DNAs. You know, 99.9% of our DNA are same. 99.9% of our DNA is same. If I take example of, let's say, what's your name? Siddharth and Sophia. I take out their DNA and I look at what are the genes which are there, what is the ATCG wala combination pairs. 99.9% of it is same. Not only between Siddharth and Sophia, but between let's say an Indian and an European. But 0.1% may there is difference, and this difference is unique for every individual. Every individual will have some uniqueness. If we use that uniqueness to identify that individual, that is DNA fingerprinting. Let's say our thumbprint is unique. In the same way, there are portions of our DNA which are unique, which is not going to be same for any other person except a special kind of twins. You know, twins can be formed in two different ways. One, if two of the mother's eggs get fertilized by two different sperms, तब भी दो बच्चे पैदा होंगे, but they are genetically different. One egg, other egg, these two eggs were different, and the two sperms which came were also different from each other. So these two are different, though there would be a lot of similarities, but they are different organisms. Another way in which twins are produced is an egg gets fertilized egg, and then it divides into two. And then it divides into two. In that case, what happens? They are exactly same. They're exactly same. When they're exactly same, their DNA fingerprint would also be same. So except in this case, we'll have this unique DNA fingerprint. Okay, we'll go into the technicalities, how DNA fingerprint is extracted and all. But plus, please understand what is the purpose of fingerprinting, DNA fingerprinting. Your purpose of your fingerprint was identifying you, you are that person. If you have gone and murdered someone and have left your fingerprints there, using that you can be identified. In the same way, if you have left your DNA somewhere, if I touch this board like this, some dry skin cells are left there. In every cell we have a DNA. From there, the DNA fingerprint could be extracted. And if, let's say, I am one of the suspect in this classroom, who touches that part of the board, I touch that part of the board. So my samples would be checked with the DNA fingerprint which are there, and if they match, I am the person. Just say, if I would have left fingerprint, in the same way I could also leave the DNA. So understood, DNA fingerprint का मतलब समझ में आया? Fingerprinting is unique identification. We'll go into the technical aspect of it, but let's first understand its applications. कोविड फैल रहा है, इतना मत खांसो, मास्क लगा के खांसो। अगर इस बैच में कुछ हुआ तो मेरी 7 दिन की छुट्टी। देखो, I didn't get COVID in 2020, I didn't get it in 2021, I got it twice in 2022. And fortunately in my case, I had fever for a day, but I was still positive, so I enjoyed for another 7 days. अपने आप को रूम में बंद करके, I was watching TV series, I was ordering food from Swiggy, and life was set. And no one was saying anything. कि तेरा वेट बढ़ जाएगा, ये हो जाएगा, सब like take care of yourself. eat healthy. For me, healthy is biryani. Okay, coming back to, so let's talk about applications. We'll go to the how DNA fingerprinting is done, but let's understand once we understand what DNA fingerprinting is, let's understand the application. Identification, that we have understood. Physically connecting a piece of evidence to a person, same, it's related to identification. Determining paternity and other relationship. It may have medical applications also. Match tissue of organ donors with those of people who need transplant. My tissues, suppose I'm an organ donor, someone who needs transplant, unme if there is similarities in their fingerprint, it would be there would be higher chances that this organ would be accepted by that body. Identify disease that are passed down through your family. Again, fingerprint can help you identify similarities there, and you can do that. Help find cure for those diseases called hereditary. Fingerprint, your DNA fingerprint will, the key characteristic of your DNA fingerprint would say, okay, you may suffer from this particular disease. There are other fingerprints, those are the people who may not suffer from those diseases. So, to find treatment for this, you can identify which are the genes responsible for it and thus treat them. Okay, these are the applications, but let us understand how the system of DNA fingerprinting came into play. There was this scientist, Alec Jeffreys. He noticed something. What did he notice? He found that there is DNA on which we have genes. Let's say this is gene 1, this is gene 2, and so on. Fine. But in between these, notice that certain sequence of highly variable DNA which don't contribute to the function of genes. So on DNA, there are some parts which are doing the function of genes, but there are some parts which are not doing the function of genes, and they are highly variable. Highly variable, जैसे जो gene होगा, मेरा gene may be similar to her gene because we are producing the same protein. 99.9% of things are same, but the non-gene part of our DNA was highly variable. It does not contribute to the function of genes and are repeated within the DNA. They are repeated, बार-बार repeat हो रहे हैं, and they are highly variable. Highly variable means that part is different in my gene when compared to her gene and her gene. So now, when we'll talk about fingerprinting, तो जो common चीज है वो तो fingerprint में काम नहीं आएंगी. What will be useful in fingerprinting? The highly variable non-genetic part of DNA. This he called mini satellites. So it was also noticed that each individual has a unique pattern of these mini satellites. The only exception being multiple individuals from a single zygote, such as identical twins. जो आपको समझाया था कि only in case of identical twins, these mini satellites may be different. Okay. देखो, there are he has a question. Sunny is the name, no? Sunny has a question. He said, then why do those twins are not exactly same? If they are formed from the same zygote and they carry the same genetic material, why are they not exactly same? अरे लगाओ, common sense लगाओ ना. The environment in which they grow, the resources that they get, all those things are different. है ना? Just that genetic material became exactly same, but the resources which they get while they are growing, one is playing, other one is not playing. One is getting a lot of vitamin D, other one is not getting. So one may have other kind of growth, other may have other kind of growth. In this way, they can be differentiated. Genetically, they would be same. Good question. Matlab, he's trying to understand how then those two people are different. Okay. Anyways, come to this. So, did you understand? On our DNA, there are some genetic part, there are some non-genetic part, and they are also highly variable. It was noticed that each individual has a unique pattern of mini satellites. The only exception here, DNA fingerprinting is a technique that simultaneously detects lots of mini satellites in the genome to produce a pattern unique to an individual, and this pattern is called DNA fingerprint. This pattern is called DNA fingerprint. Non-coding part of DNA. Fine. How is DNA fingerprint created? First, sample of cells are taken, then the DNA is identified from them, extract and purify DNA from these cells, extract DNA, then make multiple copies of it. Polymerase chain reaction is a mechanism by which if you have some pieces of DNA, you can make multiple copies of it, and then using various biomolecular sequencing mechanism, identify the satellite sequence in there, and those satellite sequence, variable satellite sequence will be your fingerprint, will be your DNA fingerprint, which would be unique when compared to any other person's DNA fingerprint. Fine, got it? हाँ, जो नॉन प्रोटीन कोडिंग वाला पार्ट है, यस. No, please understand this. Now he has a doubt. Sir, we have 3 billion base pairs. Now all those 3 billion base pairs are not contributing to, please note, are not contributing to some information encoding. Some of it is non-coding part also. Some of it is not. हाँ, वो नहीं होगा, right? Yes. Right. In fact, we would study in sometimes what is called dark DNA. Dark DNA is that portion of DNA which we have not even been able to sequence. When we take a DNA, there are parts we say gene 1 is producing this, gene 2 is producing this. This is the satellite portion which is not doing anything. But there are portions of DNA which our current sequencing mechanism have not been able to sequence also. So we still do not know exact genetic structure of human beings or any other living organ. We'll explain you what dark DNA is, what is the concept of dark DNA, but सब में होता है. We'll come to that. रुको तो. देखो, अगर humans के genes को हमने sequence कर लिया, तो एक चूहे का भी कर लेते ना. If we have not been able to sequence a चूहे का gene properly and there are dark DNA, so human genes may also have dark DNA. Common sense. पर होते हैं पक्का ना, मैं आपको एक्सप्लेन करता हूँ. हाँ, they are repeated within the DNA. Those mini satellite portion, yes, they are repeated there. Okay. No. Any cell. See, in our wait. In our body, only RBC does not have nucleus. Your skin cell, your heart cell, your eye cell, all those have nucleus. RBCs don't have nucleus. The skin is also useful. He was just saying, you see, sir, RBCs don't have nucleus, but WBCs have. Yes, WBCs have nucleus. They will have DNA. In the same way, your skin cell will have DNA. In your school, you must have done that experiment, no? You would have taken out a strip of your cell, skin, and then उसे microscope के नीचे रखा होगा. क्या था? और you must have done it for onion peeling वाला किया था. Okay. My teacher was more cruel. We did the onion वाला thing, and then she chose few students. तुम तीनों समझदार हो, धीरे से निकालो. And then she made us use this peel also. Anyways, did we understand DNA fingerprinting? Okay. Species, can we identify species using genetic? His question is, sir, can we produce artificial DNA? Can we produce artificial DNA? Theoretically, yes, but practically, right now we are at very basic level. What we have done is very basic living organisms who have very few base pairs. We have removed their nucleus and we have added our own system of base pairs, our own system of genes in them, and scientists are studying how they function. ह्यूमन बींग्स में वैसा भी हुआ नहीं है, थ्योरेटिकली दैट कैन बी डन, यस ऑफ कोर्स. अगेन थिंक ऑफ दैट, नो, सिंगल सेल which has a nucleus which is fertilized, which is going to multiply, you change that nucleus altogether. That's possibility, we'll come to that. एक क्लास में 3 डाउट का रूल पता है ना, दूसरा है, पूछो. हाँ, we'll come to that. No, you are going to designer baby वाला idea. अभी तो we are into the basics. We'll come to the concept of designer baby also. We'll come there. Okay. DNA fingerprinting is understood by everyone. Okay. Let me go to. There's a problem also. See, for DNA fingerprinting to function properly, the system has to be very foolproof. Any contamination, any kind of problem at the time of analysis and all could add to errors. And therefore, in judiciary, getting DNA, there are various reasons by which DNA fingerprint may or may not get accepted. Okay, now let's come to DNA barcoding. Now, the core difference between DNA fingerprinting and DNA barcoding, core difference is this. In DNA fingerprinting, we were identifying a unique organism. My DNA fingerprint would be different from her DNA fingerprint, from his DNA fingerprint, from her DNA fingerprint. In DNA barcoding, we are identifying the species, not the individual. Individual को identify करने के लिए what are we using? Fingerprint. In barcoding, what are we doing? We are identifying the species. Right? Suppose, abhi recently there was this thing in news. Scientists developed a mechanism, a barcoding mechanism to understand whether we have invasive fish species in our water bodies or not. So to understand whether there are invasive fish species in our water bodies or not, one way is you catch the fish species and see, invasive African catfish is here. Other is you take some samples from the soil and from water, some biological samples, some skin and all those things which may be floating in there. And then those skin will have some genetic material. You use DNA barcoding to identify whether that skin cell was part of that particular species or not. So first understand the core difference between fingerprinting and barcoding. Fingerprinting mein you are identifying the individual, in barcoding you are identifying the species. This is the core difference. This could have helped you solve last year's question, बहुत basic level पे. कि species, you will see that other two options was about identifying species, one option was ऐसे time pass. Okay, let's talk about this. How does it? So DNA barcoding is a tool for rapid species identification based on DNA sequence. It uses a short section of DNA from a specific gene or genes. यहाँ पे when we say from a specific gene or genes, we are basically saying gene sample. एक gene में तो DNA नहीं होता ना, understand वैसे वाला. So they are basically saying gene sample. So the way barcode, in the way you can using barcode in a shop identify the product. When you scan that barcode, you get information about the product. In the same way, genetic barcode or DNA barcoding can help you identify the species. Now, different gene regions are used to identify different organisms group using barcoding. Now, please understand this. See, features of a living organism or features of a species which differentiate it may be lying in different section of their DNA. Acha, before that, let me also explain you something else. We said that DNA is found in the nucleus of a cell. DNA is found in the nucleus of the cell, but there are some DNA which are also found in mitochondria. Very soon we would be studying about these basic things, and we will study mitochondria also. This is mitochondrial DNA. DNA which is found in mitochondria is mitochondrial DNA, and it only comes from mother. हम वो भी समझते हैं आज, a 3-parent baby. There again we'll explain this. So there are some DNAs which are also found in mitochondria. Then animals have what is called chloroplast. Do you know what chloroplast is? जिसमें chlorophyll है. Oh, sorry, plants. I'm sorry. Plants have what is called chloroplast, right? Chloroplast also behave somewhat like mitochondria, as they have their own DNA. So now, when we are identifying a species, there may be different regions of DNA which may be more useful for uniquely identifying a species. For example, you will see here for animals, when we are saying animals, we are including animal kingdom. We have like this ki life on earth can be divided into 5 broad kingdoms. We have Protista, we have Monera, we have Protista, fungi, plant. अच्छा, कॉम्प्लिकेटेड नाम नहीं, सिंपल प्लांट लिख देते हैं, सब मिट जा रहा है. मोनेरा, प्रोटिस्टा, plants and animals. Animals can further be divided into vertebrates and invertebrates. Vertebrates are the ones which have this backbone. So mammals, we are mammals, birds, reptiles, fish, amphibians, they're all vertebrates. Then invertebrates में the animals without backbones, so mosquitoes and all these things are invertebrates. अरे रुको देखो, यहाँ डॉक्टर्स भी बैठे, उनको और बहुत ज्यादा पता है, सब बताने लग जाएंगे अपने आप से, तो कैसे होगा? जितना चाहिए, एज वी सेड, जितना चाहिए हमें, हम सिर्फ उतना पढ़ रहे हैं, राइट? अभी वो ये पढ़ेंगे हम बायोडाइवर्सिटी में. टुडेज क्लास इज नॉट अबाउट बायोडाइवर्सिटी, वी विल स्टडी वेरियस एनिमल किंगडम, प्लांट किंगडम एंड देयर डिटेल इन बायोडाइवर्सिटी वाले सेक्शन में. तो वी आर सेइंग दैट एनिमल्स, फॉर एनिमल्स it may be but vertebrates, it may be invertebrates. For animals, to barcoding के लिए which part of the DNA do we use? We use mitochondrial DNA. Mitochondrial DNA में भी एक COI part है, we are not going to its detail, which is used to identify the unique species. Similarly, for land plants which are found in land, we use 2 different chloroplast gene, MATK and RBCL. Then for fungi species, we use ITS region of the DNA. But we do not have to go into the technicalities. What I am trying to say is different sections of DNAs may be able to uniquely identify a species, and that may be used. Now, ultimate goal of DNA barcoding is to create a database, to create a huge database where for every species we have a barcode. Right now, DNA barcoding can be done in many different ways. We can have tissue samples, we can have bulk samples, we can have eDNA samples. Tissue sample is something as simple as एक living organism है ना आपके पास, उसका sample ले लो, that is tissue sample, and from there you create a barcode. किसका barcode ये? This is what uniquely identifies it. Bulk sample is you go to a water body, you use a large net and catch a lot of living organisms or their body parts or their remains. From there you separate them and extract barcoding. There is another thing called eDNA, environmental DNA. Environmental DNA में you use water and soil, and वहाँ in there the basic things which are basic remains which are there, using that you are creating the barcode. So the core difference between bulk sample, this was the bulk sample wala mechanism, and eDNA is that bulk sample usually provide a large quantity of good quality DNA, which may not be the case with environmental DNA. Environmental DNA में what are we doing? You are going to water body, and from water you उस water body में dead animals का या किसी चीज के छोटे छोटे pieces होंगे या छोटे samples होंगे. We use that thing for creating barcode. Okay. Now, application of DNA barcoding. First is identifying plant leaves. When you do not, you just have a small leaf, you don't have anything else, and you have to identify this leaf belongs to which species. It can be done. Identifying pollen. Pollen, when let's say एक butterfly आपने catch किया, on the butterfly you have a small pollen. It belongs to which species? We do not know. It can be done using barcoding. Identifying insect larva. See, there are various insects. Their larvae look very similar. Larvas may do not have a lot of feature which can differentiate, by which you can differentiate larva of one species from other. But barcoding, they'll have genetic material. Barcoding can help you identify that. Investigating the diet of an animal based on its stomach content. In the stomach of an animal, there are various species that I may have eaten. Like if I'm a donkey, ये वाला घास खाया, ये वाला घास खाया, तो identifying what are the food items that I'm eating, what are the species of plants that I consumed, what are the species of animals that I consumed. From that small sample, I can take barcode and identify. DNA barcoding, is it understood? DNA barcoding is understood? Okay. Now, chromosomes तो हमने समझ लिया था. Chromosomes we get from our parents and all. Let's talk about RNA. We'll briefly discuss RNA. Before that, let me take this doubts online. Namrata Mohan, mature RBCs are only cells with no nucleus. Immature RBC does, but later lose it as their main job is to carry oxygen. It makes more space for oxygen to be carried. Okay, RBCs don't have nucleus. That's what we are saying. And Namrata ji is saying that initially they have, but when they mature, they don't have. Fine. When they mature, RBCs don't have nucleus. Mini satellites also contribute in. Mini satellites are the variable portion which identifies an individual, but they don't play any role in my characteristics. My characteristics would come from the genes. Sir, those who were not student of Pre-Cum-Mains batch were at any disadvantage, or we need to join? No. No disadvantage. Target Pre में we are starting everything from zero. Target Prelims batch, here anything that is needed for preliminary examination would be covered from zero. What is meant by non-invasive approach? Non-invasive approach is you are not taking a living organism, extracting DNA from there. You are dependent on a sample which is taken from outside. So does genome include? See, the genes which are responsible for coding protein, which are responsible for your key features, they do not have anything to do with mitochondrial DNA. Mitochondrial DNA has a completely different function. We will talk about it very soon when we are discussing cell wala section of your basic concepts. Second or third class mein, tomorrow or day after tomorrow, we'll be discussing that. Okay, let's come to RNA. So, to understand RNA, some basic things we already understand. You remember DNA? DNA had the double helix structure. DNA had this double helix structure where we had base pairs. Base 1, A, T, C, G, like this. We had these base pairs. Now, one key difference between DNA and RNA that we said was that it doesn't have T. It has U. First was this. Second, RNA need not be found in this double helix structure. RNA need not form the base pair. RNA may be single helix with base pairs. RNA may be like this, or RNA may be, in reality, it can be found in many different complex forms. So RNA stands for ribonucleic acid. Ribonucleic acid because जो अपन deoxyribonucleic acid DNA में the sugar that was there was deoxyribose. Here we only have ribose sugar. इसलिए it's ribonucleic acid, not deoxyribonucleic acid. So RNA stands for ribonucleic acid. It is a molecule with long chain of nucleotides. Long chain of nucleotide that we see. What was nucleotide? Nitrogen base, sugar, phosphate. Long chain of nucleotide RNA में भी है, but they may not necessarily form pairs with the base pair. base may not necessarily form pair. It may or may not form pairs. Like DNA, RNA is also vital for living organisms. It comes in a variety of different shapes. Unlike double stranded DNA, RNA is a single stranded molecule in many of its biology. In many of its role, it is a single stranded molecule and has a much shorter chain of nucleotide. Unlike DNA, DNA can have much larger number of nitrogen bases nucleotides. It would generally have much smaller number of nucleotides. However, RNA can be complementary. RNA can by complementary base pairing form intra-strand as well. Please note it. It may, जैसे DNA में तो base pairing होता ही है, RNA can form can have complementary base pairing in some cases. Now, what are the functions of RNA? तो देखो, in some living in some viruses which are RNA viruses. RNA viruses में what happens? Genetic material stored how? RNA. They do not have DNA. They do not have DNA. So how is genetic material stored? In the form of RNA only. So first function of RNA, not in human beings, but in the various kinds of life forms in viruses, is storing genetic information. In human beings, what is storing genetic information? DNA. In human beings, DNA is storing genetic information, but in some RNA viruses, RNA forms genetic information. So first function of RNA is it forms genetic information. Then second and more important function for humans of RNA is production of protein. Let's come to that. Let's understand this, how protein is formed. Now to understand how protein is formed, कुछ basic चीजें हमें पता होनी चाहिए. This is cell. This is a cell. It has nucleus. Nucleus has a DNA. Nucleus has on DNA we have genes. Now ये गेट किसने बंद कर दिया? इसे खुलवा दीजिए यार, हवा आती रहनी चाहिए. तो come back here. Cell, nucleus. Nucleus has what? Gene. Gene is encoding. Gene is the code for protein. Gene is the code for protein, right? But how is protein formed? Protein is formed by protein factory called ribosome. Ribosomes are lying outside nucleus. Nucleus के बाहर एक और organelle होता है cell में, which is endoplasmic reticulum. On endoplasmic reticulum surface, please, like for those of you who do not understand what endoplasmic reticulum is, we will cover it. Give me 2 classes. By then, your entire biotechnology, health and basics of biology would be done. So we would cover endoplasmic reticulum in the classroom only. But for now, understand, nucleus के बाहर एक organelle है endoplasmic reticulum. On its surface is lying ribosome. The ribosome is what? Ribosome is factory of protein. Factory of protein. Now, what happens is, ribosome is doing what? Ribosome gets an order. I want this kind of protein. Ribosome will assemble that protein and thus produce that protein. But where is the code for protein? Inside the DNA. Now, DNA doesn't come out. DNA has the code. Gene doesn't itself come out. We keep gene confined inside the nucleus so that it remains protected, it does not get modified. The information would come out on what is called mRNA, messenger RNA. Messenger RNA will bring information from DNA to ribosomes. Okay, so ये ribosome है, let's say, messenger RNA brought information about what protein has to be formed. It got the order, I want this protein to be formed. Messenger RNA is the factory where protein would be formed. Messenger RNA is the factory where protein would be formed. But who's getting the raw material? How is protein formed? Protein is basically a chain of amino acids. Protein is chain of amino acids. Messenger RNA brought the order, I want this protein. Ribosome will make that protein. Raw material में क्या आएगा? Amino acids are brought by another kind of RNA called transfer. tRNA will bring amino acids to it. It will take amino acid, from another tRNA, it will take another amino acid, and thus form chain of amino acid, which is protein. It will form chain of amino acid, which is protein. So look at this figure. How is protein getting formed? See, this is nucleus. Inside of the nucleus, nucleus के बाहर information कौन लेके आया? mRNA. It went to the ribosome. ये ribosome है. It went to ribosome, got attached to it. Now ribosome has to form a chain of amino acids. So who brings amino acid to ribosome? Transfer RNA, tRNA. It brought amino acid attached to it. Then tRNA is released. tRNA will go and get attached to another amino acid, and it will keep going there, and protein would be formed. So in protein formation, RNA played a role. Messenger RNA, transfer RNA, they all played a role in protein formation. So we were talking about the functions of RNA, roles of RNA. One, they can store genetic information in some RNA viruses. Second, they play an important role. They play an important role in the manufacturing of protein. The main job of RNA is to transfer the genetic code needed for the creation of protein from the nucleus to the ribosome. The process prevents DNA from having to leave the nucleus. This keeps the DNA and genetic code protected from damage. Without RNA, proteins could never be formed. This will help you understand RNA interference technology as well. In 2019 preliminary examination, a question called on RNA interference technology was asked. Now see, messenger RNA is carrying the information. He formed this protein. Though you have gene inside, gene is encoding the information. Who is carrying the information? Messenger RNA is carrying the information. Now, what if we stop the messenger RNA from functioning? In spite of the fact that you will have that gene, that protein will never be formed. Even if you will have that gene, the protein will never be formed if you interfere with messenger RNA. This is RNA interference. It is used for gene silencing. A gene is there in your body, but it is still silenced because we are not allowing it to get expressed. That is RNA interference. We will come to RNA interference technology. अभी उसे और detail में समझते हैं. Got it? Now, some RNAs also act as enzymes. So some RNA enzymes are called ribozymes. So RNA storing information in RNA viruses. RNA plays an important role in manufacture of protein, and then RNAs can also act as enzymes. Those are called riboenzymes. Okay, so messenger RNA, tRNA, do we understand this? Then there is another RNA called rRNA. rRNA is basically, ribosomes are made up of this RNA. These the ribosomes are made up from proteins and ribosomal RNA. उनको हम rRNA बोलते हैं. So ribosomes themselves are made up of rRNAs. So mRNAs are bringing the information, tRNAs are bringing the amino acids, and ribosomes themselves are made up of rRNA. See how significant RNAs are for genes to function. Genes would not be expressed if RNAs are not there. Fine. Now, where is RNA formed? How is RNA formed? RNA is formed from DNA by a process called transcription. और डिटेल में नहीं जाना है कैसे फॉर्म होता है वो नहीं. What is transcription? Transcription is the process of forming RNA from DNA. RNA from DNA. That is transcription. इतना बस ध्यान रखो. ठीक है. There is another term that you should know, transcriptome. Transcriptome. Please remember transcription because अभी हम जब RT-PCR करेंगे, reverse transcriptome, तो you should understand transcription is what? DNA से RNA बनना. Reverse transcription में RNA से DNA बनेगा. So in your COVID test, RT-PCR is done. What is PCR? DNA samples की multiple copies बनाना. That is PCR. But in COVID-19 virus, we do not have DNA. We have RNA. So first, what is done? RT, reverse transcription. From RNA, we make DNA, and then PCR, multiple copies of that, and then that is studied. RT-PCR tests. Abhi, we'll understand RT-PCR test in more detail. But did you understand transcription? Now, what is transcriptome? transcriptome basically represents all the genes in your body which are going to get expressed. My body has 20-25,000 genes. Human body has 20 to 25,000 genes. We had just studied that. We have around more than 3 billion base pairs, 20 to 25,000 genes. Now, many of those genes may not get expressed. Why? We may not have the relevant messenger RNAs. So, transcriptome is the set of all messenger RNA molecule in one cell or a population of cells. Transcriptome will basically tell which are the genes which are going to get expressed, because only if you have messenger RNA, those genes are going to get expressed. So, if you have a cell, transcriptome of that cell will be all the messenger RNAs which are there in the cell, will basically be all the genes. Messenger RNAs are playing a role in expression of the gene. So, they are basically telling us what are all the genes which are going to get expressed. If you have a population of cells, not one cell, if you have a population of cells, then their transcriptome will be messenger RNA of all the population, all the cells together. In some messenger RNA may be there, in some other cell, some other messenger RNA would be there. So, in that entire population, transcriptome would be all the messenger RNAs from different things together. So, we will say that in this entire population, because these messenger RNAs are there, these are the genes which would get expressed. Do we understand what transcriptome is? Pakka? Okay. Because transcriptome includes all messenger RNA transcripts in the cell, the transcriptome reflects the genes that are being actively expressed at any given time. Got it? So, these were some basic things about biotechnology, about how our body functions, and इसी में changes जब हम करेंगे, will come to modern biotechnology. So, the next topic that we are going to start is genetic engineering. How these DNAs could be engineered, how these DNAs could be modified. Okay, so before we go into that, so this is clear, no? Process of manufacturing of protein, do we all understand? Nucleus, inside the nucleus we have DNA, on the DNA we have genes. The information related to those genes are brought outside the nucleus to the factory of protein by messenger RNA. Messenger RNA brought it outside to ribosomes. Ribosomes will read the code that is needed and accordingly will pick up amino acids from various transfer RNAs. Ribosome itself is made up of rRNAs. Ribosome is made up of proteins and RNAs. So, ribosome itself is made up of rRNAs. Fine. So, this we understand. Okay. RNA interference technology से भी समझ लेते हैं. RNA interference. आप ऐसे क्यों बैठे हैं, सीधा क्यों नहीं बैठे? चेहरी वैसी है. अच्छा, बताइए बताइए. मैं आपका नाम भूल गया. नीरज. हाँ, नीरज जी. हाँ, RNA देखो RNA itself is capable of transferring information, no? वो रहेगा ना, उनमें रहेगा. देखो कई बार क्या होता है ना, प्लीज अंडरस्टैंड दिस, समटाइम्स देयर आर डिफरेंट काइंड्स ऑफ वायरसेस, पॉजिटिव सेंस, नेगेटिव सेंस, इस तरह की चीजें. RNA viruses can also be of different types. There are some RNA viruses where the RNA itself can be used for manufacture of the protein. जो genetic code store कर रहा है ना, that itself can be used for sending information for manufacturing of protein. The function which is performed by RNA in the human body is performed by RNA in the virus. Yes, DNA storing the information that was stored by RNA in viruses. DNA है नहीं, वो भी RNA करेगा. Yes. So Neeraj has a question. Neeraj is basically summarizing this. Neeraj is saying, sir, because RNA viruses do not have DNA, so what is the function of DNA? Storing genetic information. So that function is done by RNA. Plus all the functions of RNA are also done by RNA. Okay, now RNA interference technology. Because we understand messenger, how protein is being formed, you will understand if you interfere with RNA, you can stop the expression of some gene. You can stop the expression of some gene. So RNA interference technology is used for gene silencing. Suppose मैं एक इंसान हूं, नहीं सपोज क्या मैं तो हूं, I'm a human being, and let's say there are some genes in my body which are leading to the problem of cancer. Those genes are, let's say, mutated and are unnecessarily creating problems in my body. RNA interference technology would do what? The messenger RNA related to those genes could be silenced. So those genes would not get expressed. My cancer gets treated. RNA interference technology can also be used for fighting some pathogen, like some bacteria or some some virus infects me. Now the bacteria that is infecting me, the virus that is infecting me, that is able to grow and multiply because it is able to make protein. If we interfere with their RNA, what would happen? They will not be able to grow, multiply and harm us. They may be containing some protein which is specifically harmful for us. We may disable that protein by disabling RNA, disabling their messenger RNA. This is RNA interference technology. The basic concept. एग्जाम में जब आ जाता है ना, तो हम सोचते हैं कि यार वी आर सपोज टू ओनली नो साइंस टिल क्लास 10. हाउ डू वी नो RNA interference? See, you only need to know science till class 10, nothing else, plus current affairs. And current में जब RNA interference is in news, barcoding is in news, fingerprinting is in news, so we will study it in slightly more detail using the concepts that we study only till class 10. So class 10 तक का science plus its application is what is part of your syllabus. You will not say the sir fingerprinting तो we study only, or barcoding we study only in class 12, or barcoding is something that we study only in graduation, so this is out of syllabus. No, the concept DNA, RNA, this we study till class 10. उसका application can be of higher standards also, and UPSC may ask it. It will not be immoral on their part. वैसे उनको morality से कोई मतलब नहीं. Fine. Sir, in chemotherapy treatment we stop mRNA working, not? नहीं, in chemotherapy treatment no, हम cancer cell को मारते हैं by the chemical treatment. There we are not using, there we are not doing RNA interference and all. है ना? RNA interference would be using biotechnology only. सर RNA का वर्क नहीं समझ में आया. अरे तो रीवॉच द वीडियो ना, थोड़ा पूरे पहले चले जाओ. The advantage of having videos are you can always go back and rewatch it. बाकी सबको समझ में आया? So if most of the students are able to understand, it means आपने कुछ miss कर दिया होगा while listening, so you can listen to that portion and that should be okay. Let's go ahead. Two core techniques that have enabled birth of modern biotechnology. See, modern biotechnology में what are we supposed to do? We are supposed to modify the living organism, और एक we should have a very neat and clean environment so that any kind of contamination would not be there. So one core thing that has made modern biotechnology possible, that is we having the ability to modify the living organism, is genetic engineering. What is genetic engineering? Gene को engineer करना, DNA को engineer करना, DNA को modify करना. So we had cotton का DNA, we modified the DNA, we introduced the gene there so that it now starts producing toxin and thus becomes pest resistant. Right? This is genetic engineering. Did we understand genetic engineering? This is also called recombinant DNA. Recombinant मतलब modified DNA, recombined DNA, recombinant DNA technology. Genetic engineering is also recombinant DNA technology. Right? Now you should know some basic terms that are, अच्छा, some basic terms that would be like, if you understand those basic terms, you will be able to read the question statement and interpret them properly. We are saying that just say when we are defining genetic engineering, we are saying techniques to alter the chemistry of genetic material. Techniques to alter the chemistry of genetic material, DNA or RNA, to bring about desired modification into host organism and thus change the phenotype of host organism. Now, we have used the word here phenotype. So basically, genetic engineering is changing the genotype, changing the gene to bring a change in phenotype. Phenotype is the physical behavior. तो हमने जीन चेंज किया so that its physical behavior changes, it starts producing toxin which was not the part of its behavior earlier, which was not part of its characteristic earlier. So which genetic engineering is change in the genotype, changing the genotype to bring changes in the phenotype. For example, jellyfish grow at night. Ab rather than having golden hairs, my father would have wanted मैं रात में glow करूं. Right? What could have been done? Jellyfish could have been chosen. है ना, उनका जीन थोड़ा ले ला के डाल दिया गया होता. हंसी आ रही है? नहीं हंसो हंसो, कोई बात नहीं. इमेजिन कर करके हंस रहे हो क्या पूरा? Now, one more thing. Do we understand what is hybridization? है ना? Hybridization में is not genetic engineering. Please note it carefully. Now see, what is hybridization? To understand hybridization, you must have seen various cows in India. जैसे there is Jersey cow. Jersey cow. Do we understand? So Jersey is a cow which is not indigenous to India. It's basically a hybrid cow. Okay, what is a hybrid cow? In case of hybrid cow, we made an indigenous variety mate with an indigenous variety of animal or cow mate with a foreign variety. We got a hybrid variety of cow. Why did we do that? Because see, the European variety of cow was producing more milk. European variety of cow was producing more milk. So why not just get the cow itself rather than creating hybrid? There was a problem. The European variety of cow cannot survive in Indian conditions. Indian tropical conditions, they cannot survive. So what we wanted was, we wanted that abilities of our cattle to survive in Indian condition and high milk production of the European variety. We wanted them. We wanted that. So we created hybrid. But when we create hybrid, it is not necessary that in the very first go we will have both like desired features from both the sides. What may have happened from Indian side, it may have got less milk producing feature, and from European side it may have got less survivability in Indian condition feature. So hybridization में it's not necessary that we may get desired features. We will have to do various experiments, or we'll have to take do this multiple times till the time we get our desired feature. This is one of the limitations of hybridization. Now in hybridization also, we are creating an organism which has genes from different kind of genes from some other animals which had specific genes which make them produce more milk, right? But this is not genetic modification. Hybridization is not considered GM crop. Hybrid animals are allowed in India, but do we allow GM animals? No, not yet. Like hybrid crops are allowed in India, but do we allow GM crops? No, other than BT cotton, no GM crop has been, no GM food crop has been allowed. अभी कुछ हुआ है किस पे? धारा मस्टर्ड हाइब्रिड 11 पे, बट उस पे भी स्टे लग गया है, राइट? तो here what we are saying is advantage of genetic engineering over traditional hybridization. Not necessarily, but generally what happens is creating a hybrid of two different species doesn't work. Doesn't work in the sense, in case of animals, we end up producing non reproductive animals. But hybridization between different species also work. think of fruits, think of vegetables. There we create hybrids which are of different species altogether, and they do work. Okay, let's come here. So traditional hybridization process used in plants and animal breeding very often lead to inclusion and multiplication of undesirable genes along with desired genes. This is one of the limitations of hybridization over genetic modification. In genetic modification, in case of genetic engineering, we ensure that specifically that gene that we wanted gets added here, gets included here. Got it? Now, so we are saying that there are two bases for modern biotechnologies. One is genetic engineering and other is maintenance of sterile, microbial contamination free ambience in chemical engineering process. This is again something that we were not capable of doing in the pre-medieval era or in the medieval or ancient era. हम क्या करते, हम किसी जगह को अच्छे से धो देते, but that dhona would not have removed all the microorganisms from there, right? So two things which have made modern biotechnology possible, one is genetic engineering and other is maintenance of sterile, microbial contamination free ambience in chemical engineering process. Without this, genetic engineering or RT-PCR or these kind of things would not be effective, would not solve the problem. Now, before we go into the details of genetic engineering, please understand some more terms. Just इनको terms के form में समझो. Do not think of how technically they function. In genetic engineering, we use various tools. For example, there is a DNA. Now, in the DNA, if we want to add some more gene, some gene from outside, we'll have to cut that DNA. We'll have to cut that DNA. है ना, तब gene introduce कर पाएंगे, and then we'll have to, after this gene is introduced, the DNA have to be repaired again. Recall चाहिए होगा. Gum would be needed. So the scissor which is used to cut the DNA is called restriction enzyme. The term restriction enzyme, when you hear the word restriction enzyme, please understand this is the molecular scissor which is used to cut the DNA. Similarly, DNA ligase, अभी हम vector पे आते हैं. DNA ligase is क्या? Ligase is the favicol there, which will be used for joining. It's a specific type of enzyme, a ligase that facilitates joining of DNA together by catalyzing the formation of a phosphodiester bond. वहाँ पे sugar and phosphate का वो जो bond देखा था, it plays a role there. Then we also have what is called cloning vector. What is cloning vector? See, think of this. We want to introduce some gene from some other living organism into winkin. कौन से लिविंग ऑर्गेनिज्म का जीन चाहोगे बेटा, बताओ? हैं? सांप दे दें आपको, नागिन टाइप? कितनी खुश है. चलो, we want to introduce some gene from snake into winkin. पक्का? ऐसे ऑफेंड तो नहीं होगे ना? खुश है ये, सर बना दो, बच के रहना तुम. How will we do that? Will we take out multiple genes from there and then we will introduce it in all her cells? No, it doesn't work like that. What we do is we take out genes from there and then we introduce those genes into a vector. Vector is carrier. That vector can be a virus. हम उसमें वो gene introduce करेंगे and then that virus would be inserted in her body. In her body, that virus would multiply and thus can create multiple copies of those genes and infect and introduce those multiple copies of foreign gene in her nucleus. So that vector is cloning vector. It is creating multiple clones, multiple copies, and it's also a vector. That vector is cloning vector. So we have understood what? First, the molecular scissor. What is it called? Restriction enzyme. Then ligase, the gum, DNA ligase, and then vector, cloning vector. Okay, and then the host organism जिसमें वो DNA डालना है, जो gene डालना है. Okay, and there are various methods that are used. You will see that techniques such as micro injection are used. Here, recombinant DNA is directly injected into the nucleus of an animal cell. The recombinant DNA is inserted into the nucleus of a animal cell. In case of plants, we prefer biolistic or gene gun mechanism. Here, the cells are bombarded with high velocity microparticles of gold and tungsten, and this gold and tungsten is coated with DNA in a method which is known as biolistic or gene gun mechanism. Okay, now genetic engineering ka the most popular tool, the most famous tool is CRISPR-Cas9. In fact, in the 2020 Nobel Prize in Chemistry was also for CRISPR. Even before that, in 2018 and 2019, UPSC had asked questions, both the years. In 2018 as well as in 2019, in preliminary examination, they had asked questions on CRISPR, very basic questions. But we would study it in more details now. Why? Because, before we go into the details, please note that CRISPR tool is one of the tools which has been awarded Nobel Prize very quickly. It was developed only 10 years back. It was developed only 10 years back, and its impact is so huge. It has simplified genetic engineering. It has simplified genetic engineering so much that the scientists who developed it were given Nobel Prize very quickly. Generally, scientists wait for 10 years, 20 years to when the impact of their finding, when the impact of their discovery, when the impact of their new tool becomes visible. In very few cases, बहुत जल्दी Nobel Prize मिलता है. One of the fastest Nobel Prize winning invention was the production of insulin. इंसुलिन आज से 100 साल पहले पहली बार डेवलप हुआ था. Before that, someone who was born with type 1 diabetes generally died in 6 months or in 1 year because insulin नहीं था. So we will study about insulin also in tomorrow's class when we are dealing with health. We will study about insulin because it has completed 100 years recently. But it was one of the few discoveries जिसको फटाफट नोबेल प्राइज मिला है. ऐसा ही क्रिस्पर को भी मिला है. क्रिस्पर को नोबेल प्राइज मिला है. कुछ तो हो गया. Okay, so what is CRISPR? First, at very basic level, what is CRISPR? CRISPR is a genetic engineering tool. CRISPR is a genetic engineering tool. Do we understand what genetic engineering is? CRISPR is the latest genetic engineering tool which is cheaper than previous tools, which is faster than previous tools, which is easy to use than previous tools, which is more accurate than previous tool, which is more versatile than previous tools. तो ये सस्ता और easy to use तो है ही, पर it is also more versatile. It can do much more things than what previous tools could do. So previously, before CRISPR was there, very few labs throughout the world had ability of genetic engineering. Very few people could do genetic engineering. But now, what has happened? Genetic engineering could be done in various parts of the world very easily because of CRISPR. Now, पहला क्लैरिटी तो ये आया, CRISPR क्या है ये समझ में आया. We will go into the details of CRISPR in some times, but please understand how CRISPR was developed. Please understand how the idea of CRISPR came into play. How the idea of CRISPR came into play. Various bacteria, they used CRISPR mechanism to defend themselves. तो CRISPR nature में exist करता था पहले. CRISPR existed in nature before. Bacteria used CRISPR to protect themselves from other pathogens, specifically viruses. What used to happen was, when a bacteria was infected, by a virus, तो virus का जो genetic material है ना, it will keep that genetic material stored in its own DNA. Bacteria will keep that genetic material stored in its own DNA, so that next time when this virus infects this bacteria, what it will do, it has a CRISPR mechanism where it will go and cut the bacteria ka genetic material, because it can identify the bacteria ka genetic material from the previous attack. Now using its CRISPR tool, it was going and cutting the bacteria ka genetic material so that they would not function. This was already going on in various bacteria. scientists were studying these bacteria and found, यार ये cutting mechanism तो is very accurate. Can we not modify it slightly and make it a genetic engineering tool? This is what exactly was done. This is what exactly happened. The mechanisms which bacteria were using to cut the genes of the DNAs of viruses were used to create a very accurate genetic engineering tool called CRISPR-Cas9. So far what we have understood is CRISPR. Did we understand CRISPR? Basic level pe. If someone asks you what is CRISPR, you should be like, it's a genetic engineering tool, I know that. Zyada gyan mat pe lo. I know at least this. But now we would understand how exactly it functions, what are its various components. जो CAS9 बोल रहे हैं, that is basically CRISPR associated protein. The bacteria had various CRISPR associated proteins which helped bacteria to make cut. The CRISPR associated protein which is used in CRISPR Cas9 is the protein 9, CRISPR associated protein. So CRISPR Cas9 is what we want to understand. Hindraj is asking, sir, what is meant by recombinant DNA? Please tell recombinant meaning. Vartika Singh has replied, recombinant DNA means recombination of DNA by putting some genes to an organism to produce desired result. Bringing in genes from different organisms, recombining those to produce DNA. That is genetic engineering का ही दूसरा नाम है recombinant DNA. Now CRISPR Cas9. You will see that what is it? I'm not explaining what gene editing is. Let's come to CRISPR. CRISPR Cas9 is a new gene editing tool which is simpler, faster, cheaper, more versatile and more accurate than the previous techniques of editing DNA and has wide range of potential applications. Now, to understand how CRISPR was developed, background, the inspiration of CRISPR. The inspiration of developing CRISPR Cas9 came from CRISPR system used by several bacteria to fight against bacteriophages. Bacteriophages are viruses which infect the bacteria. So this was what are we saying? The idea for it came from CRISPR system used by several bacteria to fight bacteriophages. CRISPR or clustered regularly interspaced short palindromic sequence. Now, वैसे तो we do not have to go into its detail, but let me explain you what this thing is. Clustered regularly interspaced short palindromic sequence. Bacteria के genetic material में ना, what they were doing, they were storing gene of the virus which they had infected, which had infected the bacteria earlier. So how were they storing it? It was again I may not be technically exactly accurate, but because that is not needed. I'm just trying to help you understand wo palindromic sequence, those terms which are being used, why are they being used. Now let's think of this. Okay, this is the genetic material of the bacteria. So it has stored some of the DNA of the virus, then some of its own thing would be there, then again some of the DNA of the virus, some of its own thing, and so on. So this sequence where virus ka DNA bhi stored ya virus ka genetic material is also being stored so that they'll be able to identify it in future is this clustered regularly interspaced short palindromic sequence. So CRISPR are short DNA sequence found in genome of the prokaryotic organisms such as bacteria. Do we all understand what prokaryotic organisms are? Prokaryotic organisms are those organisms which have specialized organelles in them. देखो नहीं उल्टा बोल दिया right, I'm sorry. देखो living organisms can be broadly divided into two types, prokaryotic and eukaryotic. Prokaryotic are the basic living organisms. So in basic living organisms what happens, they have a cell but they do not have specialized organelles within. For example, they will not have a nucleus. They'll have genetic material, but that genetic material would be inside the cytoplasm there would be no membrane bound organelle. In the same way, they will not have mitochondria. Membrane bound organelles nahi honge. They will not have a endoplasmic reticulum. The functions related to them may be, this specialization has not developed yet. In eukaryotic organisms, you'll have this specialization. So these are more advanced organisms. तो हमने अभी लाइफ फॉर्म्स की बात करी थी ना, मोनेरा, प्रोटिस्टा, फंजाई, एनिमल, प्लांट्स. तो all the prokaryotes are found in monera only. सबसे basic living organisms. Okay, now come back to this. So there are short DNA sequence found in the genome of prokaryotic organisms such as bacteria, which are reminders of various bacteriophage attack. CRISPR is reminder of previous bacteriophage attack that the bacteria successfully defended against. CAS9 enzyme, part of bacteria's defense mechanism, uses these flags to precisely target, cut any foreign DNA, thus protecting the bacteria from future attacks by similar bacteriophages. Please understand, CRISPR, storage of the previous attack. Previous attack जिन बैक्टीरियाज ने, जिन वायरसेस ने किया था, उनका इंफॉर्मेशन स्टोर्ड है. नाउ CAS9 is the protein, is the enzyme which will in future, if an attack takes place, using this information, go and cut those DNAs, so that go and cut those genetic material so that the attacking virus is not able to function. This is how bacteria protected themselves. So CAS9 enzyme was seen. Okay, this is a precise cutting tool. It is a precise cutting tool. CS9 is a precise cutting tool. है ना? CRISPR, the CS9 was part of this CRISPR system, which was very accurately doing genetic engineering, which was very accurately making the cuts. Using this mechanism, scientists developed CRISPR CS9, two of them, Emmanuel Carpentier of France and Jennifer Doudna of US. I may not be pronouncing their name correctly because I've mostly read them in newspapers, so I do not, while reading it's difficult to know the exact pronunciation. So basically they also do not pronounce my name properly. ना, वो मुझे संतोष बुलाते हैं क्या? No, there is a Santosh. So I will also not pronounce their name properly. ये क्या बोल रहे हैं? बोलो बोलो, बताओ बताओ, कुछ तो बताया. अच्छा, रिपीट कर रहे थे. चलिए, तो these two scientists have won Nobel Prize in Chemistry in 2020 for developing CRISPR. Please note that CRISPR was not exactly developed by only these two people. CRISPR जो ये डेवलपमेंट है, इसमें बहुत सारे लोगों ने कंट्रीब्यूट किया है, but still the tool that is being used was developed by them, because how bacteria are working and how they are defending themselves, that study was going on since last many decades. The CRISPR system was also understood in 1980. No, not the CRISPR system. The palindromic sequence was known in 1980s. They did not understand what is the purpose of this palindromic sequence, but it was known in 1980s. So a lot of people have worked on it to finally develop this new system. Now, how does CRISPR CS9 work? अब because this video is going on YouTube, मैं ये वीडियो चला दूंगा, कॉपीराइट आ जाएगा, तो आप लोग अपने आप से ये वीडियो एक बार देखेंगे. I'll explain you how CRISPR CS9 functions. The first task is to identify the gene जहां पे आपको कट करना है. Basically it's genetic engineering tool. The first task is to identify a particular sequence of gene that is cause of the problem and thus has to be deleted. Okay, so once that is done, once we know, जैसे suppose some modification has to be made in my genes, so first we'll have to identify the gene which is causing the cancer, let's say, right? Once that is done, an RNA molecule, तो CRISPR का एक जो key component है, it is guide RNA. CRISPR has two major components, two key important components. One is CAS9, which is the scissor, and another component is guide RNA. These two things make it very accurate. So what is guide RNA? Guide RNA, it's a RNA molecule which will go and get attached to the DNA sequence जहाँ पे आपको accurately cut करना है. It will guide the CAS9 where the cut has to be made. So if you'll come to this image, you see here, Suppose this is the target sequence where you have to make the cut. First what happens? Guide RNA comes and attaches here. This guide RNA is programmable. You can program it to say which is the place where you have to go and get attached. You can accurately program it. And then comes the role of CAS9. CAS9 will come here, will make the cut, and when this once the cut is made, you can make the modification here to make the mutation happen. So the two components of CRISPR, one is guide RNA. An RNA molecule is programmed to locate the sequence of DNA strand. Which sequence? Jahan pe the target sequence, just like find and search function of a computer. This guide RNA will be scanning the DNA and will find or search for that place where the modification has to be made. And then after this, a special protein called Cas9, CRISPR associated protein 9, which is often described as genetic scissor or molecular scissor, is used to break the DNA strand at specific point so that bits of DNA then can be added or removed. It will make the cut. This makes it very simple and accurate. This makes the entire system simple and accurate. CRISPR Cas9. Okay, एक बार ये कट होता है तो वहां you can go and make changes. Now, did we all understand this? First, guide RNA attaches itself to the target, then Cas9 comes and makes the cut, and then you can make the changes there. Application, the application, whatever is the application of genetic engineering, CRISPR has that application. You can create genetically modified food crops, you can fight diseases, you can have its environmental application. So we will talk about application of biotechnology. In all those places, it will have application. In fact, some of the recent examples are these. You will see curing disease which are genetic in nature. Genetic diseases को cure कर सकते हैं. Suppose because of some mutation in my genes, I'm suffering from some disease. What CRISPR can be used? What CRISPR can be used for? The CRISPR can go and remove that gene and add the healthy gene there, right? This is an example of this is how disease could be fought. See a real-time example. Several therapeutic interventions using CRISPR for disease like thalassemia or sickle cell anemia have gone into clinical trials, especially in USA. USA was already using CRISPR to treat various genetic diseases. Initial results have been flawless. For example, this is the famous case of this teenager Victoria Gray. She was suffering from sickle cell anemia. Do we understand what sickle cell anemia is? क्या होता है sickle cell anemia? Yes, and they create problem. So again, कल health वाला chapter पढ़ेंगे हम, health वाला section पढ़ेंगे. There we'll understand what sickle cell anemia is in more detail. But this is a genetic disease. So using CRISPR, she was treated, and this is seen as a landmark case where in future sickle cell anemia जैसे diseases को treat किया जा सकता है. In fact, Government of India also in 2021 have launched this 5-year project where the goal is to develop a treatment for sickle cell anemia using CRISPR mechanism. So already a lot of things are happening. Deformities arising out of abnormalities in gene sequence can also be taken care of. क्या होता है, कई बार लोग stunted रह जाते हैं, उनका growth suffer करता है because of some genetic mutation, or speech disorders may also be there because of genetic mutation, that can be taken care of. Developing genetically modified crops or animals. Already Japan has approved a commercial cultivation of tomato variety that was developed using CRISPR based intervention. Using CRISPR based genetic tools, this was developed. They are already commercially producing tomatoes which are genetically modified and which were developed using CRISPR. Okay, but there are some challenges, some limitations which has come up because of CRISPR. One is potential of misuse. Now it's so easy to use, it's so simple to use. What if terrorists get access to it? They take let's say some virus, COVID-19 only. They take COVID-19, it's highly contagious, but they make it more dangerous as well, and then they release it in population. इतना पावरफुल टूल बुरे लोगों के हाथ में भी तो लग सकता है ना. So this becomes a challenge. Designer babies. Now what is the idea of designer babies? सी अगेन मेरा ही सोचो, गोल्डन हेयर्स, दैट्स अ डिजाइनर डिमांड, राइट? That's something that is irrational, that is something which is immoral, that is something which is violating my right to life, because I'm not taking my decision on how I would look. But my genetic features have been modified to create the feature which my parents would have thought will be better. This is designer babies. Designer babies are babies in which gene changes would be made in such a way that parents would have wanted, I want this feature in my baby, I want my baby to have brown color, I want my baby to have some XYZ things. So making genetic changes in there to create, to bring those changes. Now this is unethical. This may also happen. A case had happened in China in 2018 where a scientist in China modified babies to remove some gene which make people vulnerable to HIV. So they have been thus protected from HIV. But that is unethical. Unethical in the sense, abhi international community has not agreed that we would make or we would allow changes. We would make or we would allow changes which are genetically modifying the living organism. तो जो मेरे गोल्डन हेयर्स का सपना, माय फादर डिड नॉट हैव एनी सच ड्रीम, ये आई डोंट नो वेदर ही नोज व्हाट गोल्डन लंगूर इज और नॉट. मुझे भी गोल्डन लंगूर यूपीएससी में आके पता लगा जब एंडेंजर्ड स्पीसीज पढ़ने लगे, है ना. तो ही डजंट हैव एनी सच ड्रीम, बट सपोज ही वांटेड समथिंग लाइक दैट, मेरी ब्लू आईज हो, गोल्डन हेयर्स हो, that would be unethical. That is something that is still not allowed. Genetic modified living organisms are not allowed. Gene therapy is something different. We'll come to that. Gene therapy is used to treat some disease where genetic changes are introduced locally. We'll come to that. That's different. एक पूरे living organism को बदल देना is wrong, because what happens is, one, you're going against the nature, but also if my initial genes were modified and now if my entire body have those genes will get transferred to my successors as well, to my children as well. So this was done in China, which was facing a major criticism. Then there are collateral damages also right now, because our understanding of biotechnology or the tools are not 100% foolproof, or we do not know if we'll remove this gene, that gene may have some other impact as well. So there may be some knock-off effects also. And then ethics of CRISPR, should we allow anything to change if God has made us like that? These are some of the criticisms of CRISPR, but again, these criticisms are not very relevant for preliminary examination. At the time of mains examination, we study them in slightly more detail. Prelims ke liye, understanding what CRISPR is, how do they function, is more important. Now we'll see some updates which have happened. एक PIB में न्यूज़पेपर PIB में आर्टिकल था, PIB में आर्टिकल था which said that CRISPR has been found to be successful even in very cold conditions. क्या in very cold conditions CRISPR would be effective? So the study has found the study was by Raman Research Institute, has found that CAS9 enzyme works as effectively at very low temperatures as well. Similarly, another article was there in November 2022 about CRISPR in the Hindu, where so far using CRISPR, what was done before this attempt, using CRISPR, gene editing tool has been used in humans to remove specific genes. Suppose I suffer from some cancer, let's say that is the scenario. Before this study, which we are going to talk about, gene editing tool was used for removing some gene to make my immune system fight against cancer more effectively. कुछ removal के वजह से भी हो सकता है. So before this, CRISPR gene editing tool has been used in humans to remove specific genes to allow immune system to be more activated against cancer. But now, for the first time, what has been done? Some gene was added. Some gene was added to make our immune system more effective against cancer. What happened here? आप यहां देखेंगे the human immune system has specific receptors on immune cells. On immune cells, there are receptors that can specifically recognize cancer cells and differentiate them from normal cells. Human immune cells have receptors by which they can recognize cancer cell and differentiate them from other cells. That is why that is how they will go get attached to it and destroy those cells. Now, these are different for every patient. These receptors on immune cells may be different for every patient. So, finding an efficient way to isolate them and insert them back into immune cells to generate a personalized cell therapy to treat cancer is key to making the approach feasible on a large scale. Basically, what we are saying is immune cell has receptors which can identify cancer cells. Now, these receptors are different for all human beings. If we can develop a mechanism of identifying these receptors and adding more of these receptors in our gene, adding more of those genes in our body which will produce these receptors, can it increase the efficiency of our immune cell to fight the cancer? Yes. So this was done for the first time. So here we are saying ki this will contribute a lot in fight against cancer and solving other problems. Then how gene therapy using CRISPR can cure cancer? December 2022 में this was in news in Hindi, in the Hindu, कि CRISPR was used for fighting cancer. यहाँ पे क्या हुआ? There was this girl Alicia who was suffering from T-cell acute lymphoblastic leukemia. अच्छा, before we go into this case, do we understand what T-cell is? है ना? It's a white blood cell. In fact, let's एक छोटा सा topic है. Before we go into this, we would do that. एक second. This one, understanding T cells in more detail. Or basically, do we understand what stem cells are? Stem cells क्या होते हैं पता है क्या? Stem cells are undifferentiated, unspecialized cells which can convert into one or more types of specialized cells. For example, think of this. I say to understand stem cells, think of this. When you were a single egg, when your life started, life of every living organism, be it an elephant or a dinosaur तो अब नहीं है, blue whale or which is the largest fish species? Blue whale is a mammal, not a fish. Shark, which shark? Whale shark. Whale shark is the largest fish species on earth. हम पढ़ेंगे अभी, biodiversity में will come to that. Please note, blue whale is a mammal. It's not a fish. Shark is a fish. है ना? Confusion हो रहा है. एनिमल्स को जब हम वर्टिब्रेट्स में डिवाइड करते हैं ना, देयर आर मैमल्स, देयर आर बर्ड्स, देयर आर फिश, देयर आर रेप्टाइल्स, देयर आर एम्फिबियंस. तो मैमल्स एंड फिश आर अलग, आर डिफरेंट. तो ब्लू व्हेल इज अ मैमल, ब्लू व्हेल इज लाइक अस. तो एनीवेज व्हाट वर यू टॉकिंग अबाउट? तो स्टेम सेल्स, तो स्टेम सेल्स में नो मैटर हाउ बिग द लिविंग ऑर्गेनिज्म इज, no matter how huge the living organism is, the life starts as a single cell, one single cell. Our life also started as a single cell. But was that cell a heart cell or a skin cell or a liver cell or a brain cell? We have various kinds of cells in our body. And neuron cells are different, heart cell is different and all, right? No, they were not. Eventually they multiplied. है ना? Eventually, later we would see various kind of those unspecialized cells converting into specialized cells. So those are stem cells. Stem cells are unspecialized cells जिनसे we see formation of specialized cells. तो stem cells और डिटेल में पढ़ेंगे अभी, बट ऐसे ही आप देखो कि ब्लड के जितने भी सेल्स है ना, they are formed from blood stem cells. Eventually in matured phase, what do you have? You have red blood cells, you have platelets, you have granulocytes, you have B lymphocytes, you have T lymphocytes, you have natural killer cells, right? These are white blood cells, these are red blood cell platelets and so on. All those are formed from blood stem cells. These are unspecialized cells, इससे फिर specialized cells develop होते हैं. So the white blood cells are the cells which contribute to your immune system. B lymphocyte, T lymphocyte, or B cells, T cells, they help you fight against pathogens. Last year preliminary examination में आया था ये. In preliminary examination, this question was asked. So let us understand this T cell first. T cells are a type of white blood cells. They are part of immune system and developed from hematopoietic stem cells or blood stem cells. Blood stem cells बोलते हैं. This blood stem cell. They are developed from T lymphocytes are also developed from these blood cells. They help protect body from infection and may help fight cancer. They are also called T lymphocytes and thymocytes. These T cells are also these white blood cells are also called T lymphocytes or thymocytes. Well, let's understand why they are called thymocytes. Once they are born from stem cells, they migrate to thymus gland where they develop. Why are they called thymocytes? These blood cells will go to thymus gland to develop. T cells derive their name from this thymus. T cells. In thymus, the precursor cell mature into several distinct type of T cells. This differentiation continue even after they have left the thymus. So this T cell will go into thymus and there it will multiply into various kind of specialized T cells. T cells would further become specialized and this differentiation would continue later. For example, T cell का एक important function है mediated cell death. And it is carried out by two major subtypes of T cells, CD8 plus killer and CD4 plus helper. CD4 cells or CD8 cells, these are all different kinds of T cells which will help you fight against pathogens, which will help you fight against diseases. T cells are also called as killer T cells or cytotoxic. This means that they are able to directly kill virus infected cells as well as cancer cells. T cells can be distinguished from other lymphocytes by the presence of T cell receptor on their surface. When compared to other white blood cells, they will have T cell receptors which other white blood cells will not have, and by which they are able to you can differentiate between T cells and other cells. So देखो इसे एक बार रीड करना समझना, but what is important is T cell is very important to fight against diseases. T cell can fight against cancer cell, T cell can fight against viruses and all, right? Now when T cells do not function properly and start attacking the good cells, वो बीमारी होती है जो हम पढ़ने जा रहे हैं. T cell acute lymphoblastic leukemia. What is it? It's a type of cancer where the T cells. Now do you understand what T cells are? Do you understand what T cells are? सब थक गए हो क्या? बस ये टॉपिक खत्म करते हैं और फिर कुछ क्वेश्चन सॉल्व करते हैं, फिर ब्रेक लेते हैं. हाँ, तो बस ये लास्ट टॉपिक है. लाइक ये व्हाट आर वी स्टडिंग? CRISPR इज द लास्ट टॉपिक बिफोर ब्रेक. तो जितना CRISPR पढ़ा है उतना क्वेश्चंस भी कर लेंगे ना. It would be better before we जब अब जो सेकंड पार्ट है that is much more important. GM crops and BT cotton, BT brinjal and all those things, those are very very important. Questions related to those have been regularly asked in the examination. Okay, but first let us finish this. What is T cell acute lymphoblastic leukemia? क्या आज पहला दिन आज कम पढ़ाएं क्या ऐसा? तो क्या? हाँ वो तो ठीक है, बट क्या? अच्छा, ब्रेक में कितने लोग भागेंगे? हाथ उठाओ. कोई नहीं भागेगा? पक्का? मजा आ रहा है? सही है, मजा आना भी चाहिए. जीवन में मजा नहीं तो कुछ भी नहीं. चलो ये तो कर ले. वी विल सॉल्व सम क्वेश्चंस एंड देन विल टेक अ ब्रेक. 10-15 क्वेश्चन सॉल्व करेंगे. बिना क्वेश्चन सॉल्व किए ना मजा नहीं आता. ये नुपुर, तुम कल से आगे आके बैठोगी. तो तुम्हें मैं पिछले साल से देख रहा हूं. तुम मुझे कल से यहाँ पे चाहिए हो आगे. ठीक है? What is T cell acute lymphoblastic leukemia? In this, it's a type of cancer cell where the T cells, which are class of white blood cells, equipped to hunt and neutralize threat to body, turn against the body, where T cells start attacking the healthy cells of the body and end up destroying healthy cells that normally help with immunity. The disease is rapid and progressive and is usually treated by chemotherapy and radiation therapy. Before CRISPR, it was usually treated by chemotherapy. By chemotherapy, we'll provide it with chemicals which will kill these harmful T cells, or radiation therapy. Radiation में some nuclear radiation would be given to it so that these T cells which are harmful for us get killed. But because these T cells are formed because of mutation in our genes. हमारे जीन में कुछ गड़बड़ी होने की वजह से, कुछ म्यूटेशन, कुछ चेंजेस होने की वजह से these T cells are formed. You can kill these T cells temporarily, but because they are there in our genes, what would happen? They will be formed again. And that is why chemotherapy or radiation therapy are not very effective. How gene therapy treated this? अभी जीन थेरेपी, क्रिस्पर को यूज करके एक जीन थेरेपी ने, जीन थेरेपी इज बेसिकली bringing in changes in genetics to treat some disease. Therapy is treatment. Gene therapy is bringing in changes in the gene to treat some disease. Here, the method that was used was base editing. What is base editing? देखो इस तरह के सवाल आते हैं, term base editing. बस थोड़ा broad idea रहना चाहिए what is meant by base editing. And if you understand the basic concepts, you will understand what is meant by base editing. Do we understand the nitrogen bases? A, T, C, G, है ना? So when we say that a gene has mutated, it means जहाँ पे A, T, C, G ऐसा कुछ होना चाहिए था वहाँ पे C, G हो गया है ना? म्यूटेशन का मतलब वही है ना, फॉर्मूले में उसके सीक्वेंस में गड़बड़ हो गई है. बेस एडिटिंग इज मॉडिफाइंग दैट टू मेक इट करेक्ट. Can CRISPR do it? Yes, CRISPR can do it. So, when a misarrangement in the sequence of nitrogen bases is edited to arrange it properly, when a misarrangement in the sequence of nitrogen bases is edited to arrange it properly, it is called base editing. David Liu of the Broad Institute, Massachusetts has improvised the CRISPR Cas9 to be able to directly change certain bases, and this was used for treating the disease that we have just talked about, T-all. In this case, in Alicia's case, her T cells, perhaps because of a misarrangement in the sequence of bases, had become cancerous. She had tried various mechanisms, but it did not work. Here, they used CRISPR mechanism to modify, to do base editing, by which they modified the T cells to make it effective. Fine, I have explained the entire process here for your own interest. Exam के लिए broad idea भी रखोगे ना, exam में they will not go into this much detail. Exam में they are always worried about broader idea, broader concept. देखो, though we have explained how barcoding functions, how fingerprinting functions, but in exam they have not asked about satellite wala concept. They have mostly asked about, do you understand what fingerprinting is? DNA fingerprinting is. What is its application? They have asked, do you understand what is barcoding? If you understand barcoding, you will know that DNA barcoding is used for identifying species. That will help you solve the question. In the same way, do you understand what is base editing? Base editing, if you understand what nitrogen bases are, you will understand what base editing is. And if you understand what base editing is, you can understand that T cells में अगर mutations होके problem हुई है, तो mutation मतलब sequence बदल जाना, उसको सही कर देना is base editing. It has been able to solve to treat these diseases. Not only this, there are other applications also. But before we go there, let us, अच्छा ये दो छोटे छोटे टॉपिक्स हैं, ये भी कर लेते हैं, बस दो छोटे छोटे. सच में ब्रेक चाहिए? अच्छा हाथ उठाओ, किस किस को ब्रेक चाहिए? चाहिए? ओके. एक मिनट रुको, शुरू मजबूरी में किया था, मजा आने लगा है. राज ठाकुर इज आस्किंग सर हाउ इज दिस डिफरेंट फ्रॉम स्टेम सेल थेरेपी। अभी हम स्टेम सेल्स पे आएंगे, विल स्टडी स्टेम सेल्स इन डिटेल, वहां पे स्टेम सेल थेरेपी समझाते हैं। चलो लेट्स टेक अ ब्रेक, पर ना छोटा से लेते, 20 मिनट का ले, है ना। इट्स 7:40, वी वुड स्टार्ट एट 8 PM अगेन, तो 8 PM तक ना वापस आके सेटल हो जाना, ठीक है। अच्छा 8 PM तक नहीं हो चलो 8:10, है ना। अरे बट 8:10 के बाद आई विल नॉट अलाउ एनी वन टू एंटर इन द क्लासरूम, ठीक है। पहले से सेटल हो जाना, वी विल स्टार्ट द सेकेंड सेशन एट 8:10। एक मिनट रुको रुको रुको। कर दीजिए जल्दी से। हाँ शुरू कर दूं? आ रही है आवाज? चलिए शुरू करें। ओके, a lot of you were asking me various doubts when I was sitting outside. अभी पूछ लीजिए, बिकॉज क्या होता है ना, सेम डाउट्स गेट रिपीटेड अगेन एंड अगेन, तो इट्स बेटर यू आस्क ऑल दोस क्वेश्चंस इन द क्लासरूम। तो अभी पूछिए, क्या पूछ रहे हैं आप बताइए। नॉट, उसमें नहीं जाते। देखो क्या होगा ना, इट वोंट गो, न्यू mRNAs मे बी प्रोड्यूस्ड, ऐसा हो सकता है, कुछ डिस्ट्रॉय हो सकते हैं, तो उतने कॉम्प्लेक्सिटीज में जाना नहीं है, इतना टेंशन नहीं लेना। और बताइए। जीन क्लोनिंग अभी अभी समझाते हैं। जीन क्लोनिंग का, जीन जैसे ना, आपके पास एक जीन है, उसकी मल्टीपल कॉपीज क्लोन्स बनाना, दैट इज जीन क्लोनिंग। सिंपल क्लोन, वेरी बेसिक। क्रिस्पर और देखो, रेस्ट्रिक्शन एंडोन्यूक्लियस इज अ सीजर, whereas जो हम क्रिस्पर सिस्टम की बात कर रहे हैं, क्रिस्पर इन इटसेल्फ इज द स्टोरेज ऑफ दैट इंफॉर्मेशन इन द बैक्टीरिया, क्रिस्पर। और जो क्रिस्पर सिस्टम है, क्रिस्पर CS9, उसमें CS9 इज द सीजर, जो एंडोन्यूक्लियस जैसा हो सकता है। रेस्ट्रिक्शन, अच्छा फिर इतना मुझे नहीं आता, इतने डेप्थ में मैंने कभी पढ़ा नहीं है। नहीं नहीं, एक्चुअली हो सकता है ना, देखो अंडरस्टैंड दिस कि हमें यूपीएससी के लिए जितना डेप्थ में जाना है, उतने डेप्थ में मुझे मेरे को आता है, उससे ज़्यादा मैं नहीं जाता, राइट। अच्छा तो चलिए, शुड वी हाँ, नहीं 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 नहीं, अपने जीन से एसोसिएटेड होते हैं। ओके, अभी उसे डिस्कस करते हैं। चलिए शुरू करें। सागर क्लास में था क्या अभी तक? मुझे तो दिखा नहीं। आप भी सागर हो ना? दोनों सागर एक साथ आ गए। जल्दी सेटल हो जाओ भाई। नुपुर, आप इधर आ जाओ। कुछ नहीं, आ जाओ आ जाओ। शुरू करें? ओके ओके, चलिए। अच्छा, there's a small announcement most of you may be knowing. Today is the last day for that 23% discount. जो लोग करंट अफेयर्स में एडमिशन ले रहे हैं, फ्रॉम January 1 to January 7, for 7 days we had this discount coupon welcome 23, जिसपे 23% ऑफ है। तो जिनको भी एडमिशन लेना है वो आज रात में 12 बजे तक एडमिशन ले लेंगे टारगेट प्री में, यू विल गेट 23% ऑफ। नहीं तो फिर कल से you'll again have to pay the entire fee. जो discount coupon that was valid only for 7 days. So I think if you'll go to the website and take admission, you'll be able to apply that discount coupon right now. Okay, so let's now come to dark DNA. We'll do these two topics before we jump on to solving MCQs. Okay, dark DNA. Do we understand what dark matter is? Dark matter क्या होता है पता है? Anyone, what is dark matter? अरे नहीं, that is black object. That's not dark matter. Matter which hasn't been identified yet. Okay. Anyone else? Anyone else? Anything else? In this space, there is, around 70% matter is dark matter. Okay. Okay. Anything else about? अब देखो, पता है, when we look at, शांत रहेंगे सब लोग, सुनेंगे। When we look at our universe and when we look at all the galaxies are held together because of gravitational force. They're held together because of gravitational force, right? But when scientists analyzed and studied the mass of galaxies and the force which is required to hold them together, they realized that this is not enough. The mass of the galaxy that we are thinking is not enough. there is more mass. We haven't been able to identify that mass yet, but for galaxies to be held the way they are held today, there must be more material, there must be more mass. Similarly, they have also said that there is other forms of energy which we haven't been able to identify. They're calling it dark energy. So dark matter or dark energy are those forms of matter or those forms of energy which we have proved that it's there in our universe, but we still haven't been able to understand them, identify them, study them. Dark matter or dark energy, but this isn't important for us. Just the understanding of dark matter was important for us. Now let's come to our DNA. So we say that this is gene 1. Let's say ये वो satellite वाला है, and so on, right? वो repetitive satellite वाला portion which is highly variable, and so on. We thought that we had identified all the genes which are there on our DNA. Our gene sequencing tools have identified all the genes which are there on our DNA. हमें ऐसा लगा था. But then scientists found various species. जैसे for example there was a rat species, desert rat. उसका जब हमने जीन सीक्वेंस किया, डीएनए में से जीन्स आइडेंटिफाई किया, we found that it does not have genes for some protein which are very crucial for its existence. When we sequenced its gene, we found that it does not have genes which are very crucial for its survival. Then how is it surviving? If that protein is not there, scientists did more study and found, okay, those proteins are there, but उसका gene नहीं है. Proteins are there, but the associated genes are not there. Now how is it possible? So we came to a conclusion that our gene sequencing tools are still not good enough. There are, on this DNA, there are genes which we haven't been able to sequence yet. There are parts, there are areas which we haven't been able to sequence yet. They are there. Their product, उस जीन से जो प्रोटीन बनेगा, that is there in the body. The way dark matter is there, its impact is visible. Galaxies are held together, but we haven't been able to identify them. We haven't been able to study them. This DNA, which we haven't been able to identify, which we haven't been able to study, which we haven't been able to sequence, but are there and whose impact is visible in the form of products, in the form of proteins, are called dark DNA. Got it? Did we all understand what dark DNA is? Idea behind dark DNA? Now, with the coming of dark DNA, so I have not included it in your notes क्योंकि ये news में थोड़ा पुराना था. What I have done is things which were not in news exactly in last 1 year, I have kept them in the topic. I will discuss it in the class so that you will have an awareness about that, but it would not make your notes bulky. At the same time, you will understand them. Now see, a dark DNA आने से बहुत सारे फायदे होते हैं. देखो, first, it gave us an understanding that we still need to improve our gene sequencing tool. We need to improve our gene sequencing tool, right? Second, a number of those genes which we haven't been able to sequence yet, identify yet, are playing a role in our body, maybe causing diseases in our body, and we have not been able to identify them. So it will help us better understand those diseases, and once we are able to find those dark DNA, it will help us better understand, fight those diseases. Fine. Third, do you understand evolution? Like we have, before being Homo sapiens, Homo sapiens have evolved from other Homo species and other parent species, right? Now, when we identify various species, or when we identify evolution of various species, the role of genes are very important. We say that okay, our predecessors were those, there is a lot of gene similarity. Our predecessors were those people, उनके predecessors वो रहे थे, उनके predecessors वो थे. Because of a lot of gene similarities, we say that. Now dark DNA would further add to clarity. Why? Those are the DNAs which we haven't been able to, those are the genes which we haven't been able to identify yet. But once we are able to identify them, once we are able to study them, it will help us better understand evolution as well. So it will help us better fight diseases, it will help us better understand evolution. It would, it has told us that our gene sequencing tool are not good enough. Got it? Dark DNA, समझ में आया सबको? Okay. Let's go to the next topic, somatic cell nuclear transfer, SCNT. अच्छा मैं ना, I do not like dictating things in the classroom. वो आपको खुद लिखना पड़ेगा, because that will consume a lot of time. If I start dictating things, तो जितना मैं पढ़ा पा रहा हूँ उसका 40% पढ़ा पाऊंगा. I experimented this once. So you have video access. तो आपको if you want to rewrite these things and if you are forgetting something, watch the videos again and do that. Yes. Yes, I am using the DNA and gene interchangeably, but correct is DNA का sub part is gene, right? But in books also, in newspapers also, these things are used interchangeably. कई बार कि genetic modification है या DNA को modify कर रहे हैं, तो genetic modification is basically DNA का modification भी हो रहा है, right? But understand, do you understand what is the difference between gene and DNA? Gene is the fundamental unit, DNA is the entire thing. No, it is the genes that we haven't been able to identify on the DNA. So that part of DNA which is still dark, which we haven't been able to study. So उसमें there are genes which we haven't been able to, when DNA is dark, when a part of DNA we haven't been able to study, so there are genes which we haven't been able to study, right? Okay. Upen Chauhan has a question. Sir, in DNA fingerprinting we map those DNA that, Abhi, why are we asking questions regarding fingerprinting? I think he must have started watching video अभी पुराना वाला अभी पूछे. चलो, I'll take it at the end of the class. Okay, let's go to somatic cell nuclear transfer, SCNT. Somatic cell nuclear transfer. Somatic cells refer to adult cells. Adult cells like differentiated from egg cells. Egg cells are supposed to multiply quickly and form a living organism entirely, right? Somatic cells are जैसे मेरी body का कोई cell ले लो. You take a cell from my body, my skin, my heart, my lungs. Those would be primarily somatic cells. If not stem cells, they are somatic cells, adult cells. Now, my life started as a single cell, which had genetic material from my parents. आप सबके में भी यही हुआ था, मतलब कोई डिफरेंस नहीं ना. Genetic material from our parents. Now, those cells multiplied. Those genetic material got copied in all our cells, except in matured RBCs, we have genetic material in all our cells, same genetic material. So you take any cell of my body, you take out its nucleus, it will have the genetic material that was there, that was given to me at the time of my birth, at the time of conception, at the time of fertilization, right? So you take any of my cell, somatic cell, take out its nucleus of a somatic cell. Now you take a egg cell. Egg cell का property क्या होगा? It will multiply quickly, है ना? You can stimulate it to multiply and create many copies. This egg cell may have a nucleus. Take that nucleus, throw it out. So now we have a cell, egg cell, which does not have a nucleus. Put this nucleus here. So now what do you have? You have a egg cell which has Santosh nucleus. right? Now you stimulate this egg cell to multiply. You will be able to create a lot of copies of my DNA. What will multiply? My DNA is going to multiply because this nucleus was the adult nucleus, right? What is going to multiply? My DNA is going to multiply. You will be able to get a lot of copies. In future, you want to create a clone of me. You will need a lot of copies of my DNA. This is how you will do it. Somatic cell, adult cell, nuclear transfer. उसका न्यूक्लियर ट्रांसफर किया आपने किसमें? एग सेल में. This is something in genetics and development biology. Somatic cell nuclear transfer is a laboratory technique for creating an ovum with donor nucleus, an egg with donor nucleus. In SCNT, the nucleus which contains organism's DNA of a somatic cell, a body cell other than the sperm or egg cell. Somatic cell is a body cell other than the sperm cell or egg cell, is removed and the rest of the cell is discarded. You will take nucleus from an adult. At the same time, the nucleus of an egg cell is removed. The nucleus of the somatic cell is then inserted into the unnucleated egg cell. After being inserted into the egg, the somatic cell nucleus is programmed by the host cell to multiply. After many meiotic divisions in culture, this single cell forms a blastocyst, an early stage embryo with about 100 cells with almost identical DNA to the original organism. So therapeutic cloning in these things in future, this may be very crucial. You want to create my exact liver. You want to, let's say आप मेरा liver दोबारा बनाना चाह रहे हैं, मेरे को ही जरूरत पड़ी. Egg cell multiplying again and again, eventually specialized form में वो living organism पूरा बन सकता है cloning वाले section में, and then liver can be created again. This is somatic cell nuclear transfer. Now before we go to the applications of biotechnology, we will first solve questions. Okay. ओके, जिसे जो भी आंसर करना चाहता है प्लीज रेज योर हैंड. क्वेश्चन नंबर 1, who wants to answer? Rahul wants to answer. D. Rahul is saying D. None of the above. The production of which of the following is an example of modern biotechnology? Curd, wine, penicillin. जो भी ट्रेडिशनल वे में बन रहे हैं, ये none of them are modern biotechnology. They are all traditional or classical biotechnology. Do we all understand it? Okay. Let's go to question number 2. इधर से आपसे, आपका नाम क्या है? नमन. नमन जी बताइए. Which of the following statement is not correct? DNA is the basic physical and functional unit of hereditary. Gene is the basic physical and functional unit of hereditary, right? DNA is not. बाकी तीनों सही हैं. DNA is the hereditary material in humans and almost all other organisms. Yes. Most DNA is located in the cell nucleus, but a small amount of can also be found in the mitochondria. Yes. Question number 3. Consider the following statements about DNA. Who's going to answer? इधर से आप आंसर करोगे? आपके पास नोट्स नहीं है? आपके पास नोट्स क्यों नहीं है? तो आपके कहाँ है? Question number 3. 3 only. C. 3 only. DNA is polymer of ribonucleotide. 1 is wrong. DNA is a polymer of deoxyribonucleotide. Ribonucleotide is a का polymer कौन होगा? RNA. 1 is wrong. In humans, DNA is only located in the nucleus of a cell. No, mitochondria में भी है. 2 is wrong. The order and sequence of base pairs on DNA determines information available for building and maintaining an organism. Yes. Gene sequence यहाँ पे. तो 3 only is the answer. Third का question number 3. Question number 4. 4 कौन बताएगा? आप बताएंगे? आपका what's your name? सौरभ जी बताइए. Question number 4. Now people who are answering it online, no, please also put the question number before your answer. Like 4A, 5B ऐसे question number भी डालिए. Question number 4 का हाँ बताइए. 2 only. DNA profiling is a forensic technique used to identify individuals by characteristic of their DNA. है ना? डीएनए ये तो सही है, ये तो डीएनए फिंगरप्रिंटिंग, डीएनए प्रोफाइलिंग इज द सेम थिंग. वन इज करेक्ट. डीएनए प्रोफाइलिंग determines a gene's functionality and position in the chromosome. Two is wrong. Gene mapping. First is talking about डीएनए फिंगरप्रिंटिंग और डीएनए प्रोफाइलिंग. सेकेंड इज टॉकिंग अबाउट जीन मैपिंग. डीएनए पे कौन सा जीन क्या कर रहा है, दैट इज मैपिंग, जीन मैपिंग. तो करेक्ट पूछा है तो ए होगा ना आंसर. आपने बी बोला. और आप देखो, इवन लाइक सम ऑफ यू मे हैव डन दीस क्वेश्चंस अर्लियर, क्योंकि इसमें से कुछ क्वेश्चंस मैंने लास्ट ईयर भी कराए हैं, कुछ नए भी हैं. बट इवन इफ दैट्स द केस ना, आप आंसर मत करिए, बिकॉज आपने अगर पहले करा हुआ है तो उसका मतलब नहीं है. बाकी सबको टाइम दीजिए क्वेश्चंस करने का, समझने का, एंड देन आंसर करने का. ओके, लेट्स गो टू क्वेश्चन नंबर 5. क्वेश्चन नंबर 5. डीएनए फिंगरप्रिंटिंग वाज इन्वेंटेड इन 1984 बाय प्रोफेसर सर एलेक्स जेफरीज. हाँ, ये तो फैक्चुअल है, ठीक है. ही हैड रियलाइज्ड दैट ही कुड डिटेक्ट वेरिएशंस इन ह्यूमन जीनोम इन द सैटेलाइट डीएनए रीजन. यस, ये भी सही है ना. ठीक है, बोथ 1 and 2. सी इज द आंसर क्वेश्चन नंबर 5 का. क्वेश्चन नंबर 6. टेक योर टाइम, गलत मत करना. क्वेश्चन नंबर 6. कंसिडर द फॉलोइंग स्टेटमेंट्स अबाउट वेरियस डीएनए रीजन. Satellite DNA regions are stretches of repetitive DNA which don't code for any specific protein. यही है satellite DNA, right? They are repetitive, they are highly variable among species. So one, second, each individual has a unique pattern of mini satellite. Yes, variability is this. That is why fingerprinting में this is used. So both 1 and 2, C is the answer. Question number 6 का answer is C. Let's go to question number 7. हाँ, सैटेलाइट वाला नॉन जीन है, पर वो हमने सीक्वेंस कर रखा है. देयर इज डार्क डीएनए जो हम सीक्वेंस भी नहीं कर पाए हैं. repetitive DNA मतलब this thing repeats again and again in the DNA. In the entire DNA, this satellite portion repeats again and again. But among, इसीलिए तो किसी भी पोर्शन आपको थोड़ा सा भी डीएनए मिल जाता है, उससे यू विल बी एबल टू आइडेंटिफाई अ पर्सन. ओके, लेट्स गो टू क्वेश्चन नंबर 7. ऐसे नहीं आंसर करना है, आपको हाथ उठाना है तब बोलना है. आपका नाम क्या है? अजिंक्य. अजिंक्य जी बताइए. डी, डीएनए फिंगर. हाँ, ये तो मैंने एप्लीकेशन समझाने के लिए. ऑल 5 आर एप्लीकेशंस ऑफ डीएनए फिंगरप्रिंटिंग, राइट? डी इज द आंसर. 7th का डी इज द आंसर. लेट्स गो टू क्वेश्चन नंबर 8. यू, व्हाट्स योर नेम? निवेदिता जी बताइए. बी. बी, 3 and 4. डीएनए बार कोडिंग, आइडेंटिफाइंग इंडिविजुअल्स नहीं, वो इंडिविजुअल को आइडेंटिफाई करने के लिए नहीं है, वो क्या है, स्पीसीज आइडेंटिफाई करने, राइट? डिटरमाइनिंग पैटर्निटी, नो. Determining species of a living organism, yes. Identifying insect larva which may have fewer diagnostic characteristic than adult, yes. Larva को से you cannot identify what the species because it has fewer diagnostic characteristic, but from its DNA barcode you will be able to identify to which species does it belong. So 3 and 4 is the answer. Answer is B. 8th का आंसर B. सबको समझ में आ रहा है? पक्का? Okay, let's go to question number 9. हाँ, पैटर्निटी में फिर आपको फिंगरप्रिंटिंग पे चले जाना पड़ेगा. देखो, बारकोडिंग में दिस लीव बिलोंग टू विच स्पीसीज इज फाइन, बट दिस लीव बिलोंग टू एग्जैक्टली विच ट्री you won't be able to do. For that you'll have to go in more detail, that is done in DNA fingerprinting. Okay, I'll understand it like this. You get a piece of like suppose ऐसे समझो कि क्या हो रहा है, बीफ वाला सिनेरियो है, some meat get collected, meat get confiscated. The sellers are saying it is mutton. The confiscators are saying it is beef. So barcoding say you will be able to take some samples and identify to which species does it belong. But barcoding say you won't be able to identify to which exactly cow it belonged. राइट, उसके लिए आपको और डिटेल में जाना पड़ेगा, दैट इज फिंगरप्रिंटिंग, डीएनए फिंगरप्रिंटिंग. ओके, लेट्स गो टू क्वेश्चन नंबर 9. 9 was asked in this year's preliminary examination. DNA barcoding can be a tool for distinguishing among species that look alike. Identify undesirable animals or plants material in processed food. Processed food में you can identify different species and thus you can identify undesirable species. But does it have any role in age? No. Gene तो age के साथ कोई मतलब नहीं है. My genetic material, my DNA when I was not even born was same as what it is today, right? So gene has nothing to do with age. So answer was 2 and 3. Answer was D. इतना difficult नहीं था. Just you have to understand कि barcoding is used for identifying species. With this the question gets done. Okay. Question number 10. Question number 10. You want to answer it? आपका नाम भूल गया, नाम बताओ. प्रशांत, बताइए. B, 2 only. The sequence of DNA which causes genetic diseases are known as dark DNA. नहीं. These sections of DNA have not been sequenced and they don't encode any protein. ये भी गलत है. They may encode protein. They may encode. In fact, dark DNA हमने आइडेंटिफाई ऐसे किया कि उनका प्रोटीन तो मिल रहा है, बट उनका जीन नहीं मिल रहा. तो आंसर इज डी. क्वेश्चन नंबर 10, आंसर इज डी. 10th का आंसर इज डी. लेट्स गो टू क्वेश्चन नंबर 11. आराम से देखो, गलती नहीं करनी है. अभी इन्होंने अगर ध्यान से क्वेश्चन रीड किया होता तो गलत नहीं होता. तो आराम से करेंगे. सबसे पहले नहीं करना है, सबसे सही करना है. टेक योर टाइम. व्हाट्स योर नेम? आदित्य, बताइए. डी. ऑल आर करेक्ट. ये तो हमने देखा, आरएनए एप्लीकेशंस, स्टोरिंग जेनेटिक इंफॉर्मेशन फॉर वायरसेस, मैन्युफैक्चरिंग प्रोटीन and also acting as enzymes. And then RNAs generally have much shorter chain, ये भी बताया था आपने, है ना. When compared to DNA, RNA number of nucleotides there are much smaller. So answer is D. Question number 11, answer is D. Fine. Question number 12. You want to answer? अभी रीड कर लो पहले ध्यान से, आपसे ही पूछेंगे. कर लिया? चलो सबको रीड करने दो. बताइए. बी, 3 ओनली. 1 and 2 बोथ आर रॉन्ग. mRNA ये दोनों अलग अलग कर रखा है, मतलब इंटरचेंज कर रखा है, राइट. क्या है आंसर? बी. 12th का आंसर इज बी. 13. क्वेश्चन नंबर 13. और ये ऑनलाइन इतने सारे बच्चे देख रहे हैं, लाइक तो मेरे इतने कम है. पसंद नहीं है क्या? लाइक कर दो नालायक लोगों. सर सही से ज्यादा गलत हो रहे हैं, दैट इज अ गुड थिंग ना. सही से ज्यादा गलत हो रहे हैं, दैट इज मेकिंग यू रियलाइज कि ऑलरेडी जो पढ़ा हुआ है उसमें भी गलत कर रहे हो, है ना. उससे एक पहली चीज और ध्यान से पढ़ना है. दूसरा, यू विल अंडरस्टैंड दिस कि even if you know things, if you don't read questions carefully, you will make mistakes, right? That's why rather than running behind hundreds of sources, एक को अच्छे से पढ़ो ताकि गलतियां ना हो. Let it be the responsibility of your teacher to run behind hundreds of sources. जो prelims compilations आएंगे ना, जो फरवरी मार्च से आना शुरू होंगे, I will go through all of them and I will see कि क्या बायोटेक से have I missed anything. If I would have missed, I will give it to you in updates. So let the responsibility of studying everything कंपाइलिंग एवरीथिंग बी माई रिस्पॉन्सिबिलिटी, आपका काम पढ़ना होना चाहिए एंड यू शुड नॉट बी वरीड अबाउट कहाँ से पढ़ें कैसे पढ़ें, बहुत मोटा सा साइंस का मटेरियल बनेगा आपका, स्पेस, एस्ट्रोनॉमी, नैनोटेक, रोबोटिक्स, सर नैनोटेक रोबोटिक्स तो छोटे होते हैं, कंप्यूटर, इंफॉर्मेशन टेक्नोलॉजी, इलेक्ट्रॉनिक्स, ऐसे बहुत सारे टॉपिक्स पढ़ेंगे हम, एंड बहुत एकदम यू विल नॉट मिस अ बुक, ऐसा होता है ना कि कोई बुक नहीं पढ़ी हमने, यू विल नॉट मिस इट, इतना बुक के फॉर्म साइज में मिलेगा आपको मटेरियल, इंपॉर्टेंट है ध्यान से पढ़ के उससे अच्छे से क्वेश्चन अटेम्प्ट करना। कौन बताएगा? अब मैं आपका नाम फिर से भूल गया, नमन, हाँ नमन जी बताइए। हाँ, फर्स्ट 1 to 4 इट इज यूज फॉर डेवलपिंग जीन साइलेंसिंग थेरेपीज, आरएनए इंटरफेरेंस जीन साइलेंसिंग करेगा, यस, तो 1 तो सही होना चाहिए, इससे वी आर लेफ्ट विथ ACD ओनली। इट कैन बी यूज्ड इन डेवलपिंग थेरेपीज फॉर ट्रीटमेंट ऑफ कैंसर, some genes may be creating problems, may be causing, contributing to cancer, we can silence those genes, तो yes है ना, 2 भी सही होना चाहिए, 1 भी सही 2 भी सही, यही बच गया। now please understand this, in science and technology or in any subject there may be questions about which we just have basic understanding, RNA interference के बारे में हमारे पास we have not studied it the way people study RNA interference in their graduation and all, they will understand various techniques and all, हमारे पास limited understanding, but by that limited understanding we may not have been very sure about it can be used for developing hormone replacement therapy or it can be used to produce crop plants that are resistant to virtual अच्छा ये तो चलो पढ़ा ही है, but ये नहीं भी पढ़ा होता, what by knowing questions in a limited way, by understanding that, by just having basic understanding of the topic, you can know 1 or 2 statements surely and by that you will be able to attempt question, you will not have just घबरा मत जाना कि यार ये मेरे को बेसिक तो आता है बट मैंने इसे अच्छे से पढ़ा नहीं है, इस वजह से क्वेश्चन को देख के घबराना नहीं है। देखो we did not even need to read 3 and 4 and our answer was done, we did not even need to read 3 and 4 and our answer was done। Let's look at the fourth, we'll go to 3 but let's look at fourth statement, it can be used to produce crop plants that are resistant to virtual pathogens क्या क्रॉप, हाँ वो भी आपको समझाया था कि when those pathogens would come we can do interference in their mRNA so that they do not produce the protein which are creating the problem, right? तो 1 2 and 4 तो समझ में आया, let's go to 3, it can be used to develop hormone replacement therapy, it can be used to develop hormone replacement therapy। now see, for hormone replacement you will have to introduce some gene which will produce those hormones, you will have to introduce them, so just interference would not be good enough। but now coming to, जैसे मेरे से अगर ये क्वेश्चन मैंने बनाया होता ना तो मैं एक स्टेटमेंट ये भी देता 1 2 3 and 4 एंड वहां पे शायद मैं मार्क कर, क्योंकि इट कैन बी अ पार्ट ऑफ दैट, फर्स्ट स्टेप could be stopping the production of current hormones, reducing the production of current hormones, उसमें interference could be used and then जो जिस हार्मोन को जिसको रिप्लेस करना है उसमें एडिशन, तो इट कैन बी अ पार्ट ऑफ दैट, गॉट इट, ओके। तो अभी व्हाट इज द आंसर 1 2 and 4 ए, ओके। क्वेश्चन नंबर 14, रिकॉम्बिनेंट, सर लाइक से क्या होगा दादा, दिल में बसते हो आप, अबे दिल में बसना तो ठीक है पर क्या है ना कि वी डू नॉट हैव अ बिग यूट्यूब चैनल और वी डू नॉट हैव बहुत सारा पैसा मार्केटिंग के लिए और सारी चीज़ों के लिए जितना बाकी लोग पहुंच पा, आपने देखा होगा न्यूज़पेपर में हम एड्स नहीं दे पाते, आपने देखा होगा हमारे एड्स मतलब पिछले 1 साल से न्यूज़पेपर्स में नहीं आए हैं बिकॉज दे आर वेरी एक्सपेंसिव। तो हाउ डू वी रीच मोर पीपल थ्रू आर क्लासेस, हम हर महीने में 2-3 क्लासेस यूट्यूब पे डालेंगे, some people would watch it and this is how they would reach us। अच्छा, आप लोगों में से कोई मार्केटिंग से आया है क्या यहाँ पे, कोई एक ने जिसने ऐड देख के आया हो, यूट्यूब देख के ना, यूट्यूब वीडियो देख के आया, किसी ने बस एडवर्टाइजमेंट आया है कि लेवल अप आईएएस का करंट अफेयर्स प्रोग्राम है उसके लिए, नहीं, ये मार्केटिंग बजट पूरा ही कचरा है ना। अच्छा, लाइक हाउ हैव यू पीपल कम, अच्छा उस टाइम जो, ओके, आरटीएफ की वाला वीडियो लास्ट ईयर, ओके ओके, फ्रेंड, अच्छा रेफरेंस से कौन का है? फ्रेंड के रेफरेंस से, सब फ्रेंड के रेफरेंस से आया है। हाँ? अच्छा निशात सर के रेफरेंस से, ओके। मतलब आप तो लेवल अप के ही हो। मार्केटिंग मार्केटिंग का कोई फायदा नहीं ना। ये हमने एक ना बड़ा बोर्ड लगाया है, यहाँ पे बहुत पैसे लग रहे हैं, हटवा दूं? ये नहीं, गुरु दक्षिणा बिना किसी चीज के, बोला उल्टा मैं। ओके चलिए कोई बात नहीं। तो दैट इज वाई अगर लाइक्स ज्यादा रहते हैं, ये चीजें रहती है ना, तो वी आर एबल टू रीच मोर पीपल, दैट इज इट, नथिंग एल्स। उसमें मतलब लाइक्स का कोई पर्पस नहीं है एज। आई डू नॉट गेट इंटरनल ग्रेटिफिकेशन, ओहो इतना लाइक आया। इट जस्ट वी आर एबल टू रीच मोर स्टूडेंट्स। एंड देखो आपको ये भी समझ में आएगा बाय एंड ऑफ अप्रैल 30 जो फी हमने लिया है हम उससे बहुत ज्यादा डिलीवर करेंगे, बिकॉज देयर इज नो अदर टीचर इन्वॉल्व, सारा एफर्ट मेरा ही है, तो इट डजेंट मैटर। बाकी बाकी टीचर, अच्छा ठीक है देखो, बाकी टीचर्स होते ना तो मुझे और पे करना पड़ता, इसका प्रोग्राम का फी थोड़ा हायर साइड पे रखना पड़ता, बट दैट इज नॉट देयर, क्योंकि सब कुछ मेरे को करना है, सारे क्वेश्चंस मैं खुद बनाता हूँ, आरटीएफक्यू के सारे क्वेश्चंस मैं खुद बनाता हूँ, ये पूरा नोट्स मैं खुद बनाता हूँ, तो वी कैन कीप फी ऑन द लोअर साइड। तो व्हेन वी हैव fee on the lower side, when we are doing this exhaustive coverage, we should be able to reach more students, and likes and shares would help us getting there. That is the idea. Okay, now अब आते हैं नेक्स्ट क्वेश्चन पे, आते हैं क्वेश्चन नंबर 14। रिकॉम्बिनेंट डीएनए टेक्नोलॉजी अलाउ जीन्स टू बी ट्रांसफर्ड। जीन्स किस किस के बीच में ट्रांसफर अलाउ कर सकता है जेनेटिक इंजीनियरिंग? अक्रॉस डिफरेंट स्पीसीज, कैन इट डू दैट? बैक्टीरिया से प्लांट में आ रहा है तो अक्रॉस डिफरेंट किंगडम कर दिया है, स्पीसीज तो छोटी सी चीज है। फ्रॉम एनिमल्स टू प्लांट्स, डेफिनेटली। फ्रॉम माइक्रो ऑर्गेनिजम्स टू हायर ऑर्गेनिजम्स। वंस वी अंडरस्टैंड कि डीएनए इज द जेनेटिक मटेरियल इन all living organisms except some RNA viruses, we'll understand कि criss crossing is possible, no problem at all, fine. Question number 15, consider the following statements about advantages of traditional hybridization over genetic modification. अच्छा एक मिनट, 13 में आपको डाउट किसमें है? क्वेश्चन नंबर 13, what was 13? RNA interference, fourth statement. It can be used to produce crop plants that are resistant to virtual pathogens. Think of this, crop plants are being infected by some virus or some bacteria. When they are infected by that bacteria, bacteria produces some protein which is harming it, or bacteria produces, let's say, virus का सोचो, COVID-19 virus, it has a protein on its surface, what is it called? Spike protein. अगर हम स्पाइक प्रोटीन का प्रोडक्शन ही रोक दे, विल दैट वायरस बी एबल टू एंटर आवर सेल? नो। तो बाय आरएनए इंटरफेरेंस वी कैन डू दोस काइंड ऑफ थिंग्स, दैट्स व्हाई वी कैन प्रोड्यूस प्लांट्स और एनिमल्स which will be able to protect them from these things. Okay, let's go to question number 15. Consider the following statements about advantages of traditional hybridization over genetic modification. कौन बताएगा? आप बताएंगी? विशाखा नाम है ना आपका, विशाखा जी बताइए। To only traditional hybridization leads to precise inclusion of genes? नहीं, traditional hybridization may lead to addition of problems. Traditional hybridization process is easy to perform. Yes, it's easier than genetic modification. Okay, तो देखो यहाँ पे क्वेश्चन इज advantages of traditional hybridization over genetic modification. That is why we are saying it's easy. Else it's not easy. When compared to genetic modification, it is easier. Got it? Okay. Question number 16. आप बताइए, व्हाट्स योर नेम? आनंद, हाँ आनंद, बताइए। ए, with reference to recent development in science, which one of the following statement is not correct? अब देखो यहाँ पे भी we'll learn something. अब हमने recent development science सारे तो देखे नहीं अभी, क्या क्या हो रहा है क्या क्या नहीं हो रहा है, but let us look at this question. Functional chromosomes can be created by joining segments of DNA taken from cells of different species. क्या हम जीन्स फ्रॉम डिफरेंट स्पीसीज या डीएनए फ्रॉम डिफरेंट स्पीसीज उनका फ्रैगमेंट लेके एक पूरा क्रोमोसोम बना सकते हैं? दिस इज द स्टेटमेंट, ये बना सकते हैं। नाउ प्लीज गो बैक टू आवर बेसिक फंडामेंटल. व्हाट हैड वी सेड? वी हैड सेड क्रोमोसोम इज दिस एक शेप थिंग ऑन विच वी हैव डीएनए. तो क्रोमोसोम्स डीएनए से मिलकर बनाए, और डीएनए को जब आप खोल दोगे तो यू विल हैव दिस काइंड ऑफ स्ट्रक्चर ऑन विच यू विल हैव जीन्स. So they are saying कि functional chromosomes which can function can be created by taking subsections of DNAs from different organisms. तो एक तो पहली बात ये नहीं किया जा सकता, अभी तक we haven't done this, but let's say we had doubts, we were not sure about it. Okay, let's look at other statements. That is the key. Let's look at because this is a very advanced thing to do. पहले आपको डीएनए बनाना पड़ जाएगा by adding a lot of genes. You will create a DNA, and from that DNA next step would be histone protein and then chromosome ka creation. Okay, so this is a very advanced thing to do. Let's go to second. Pieces of artificial functional DNA can be created in the laboratories. This is a less advanced thing. मतलब पहले डीएनए बनना पड़ेगा. तो if this is there, this must also be there. This is the preliminary requirement. A piece of DNA taken out from an animal cell can be made to replicate outside a living cell in laboratory. Yes, this is obvious. DNA, PCR, polymerase chain reaction, this is what we do. A piece of DNA is made to replicate, multiply. In polymerase chain reaction, this is what we do. So this is definitely correct. Cells taken out from plants and animals can be made to undergo cell division in laboratory petri dishes. That is also, we know, in if we provide nutrition, even outside our body, our cell can be made to multiply, made to grow. So C and D are definitely correct. B is a prerequisite for this. तो इन दोनों में से कोई सही होगा तो क्या होगा? पहले ये होगा ना, ऐसा तो नहीं हो सकता this would happen and then this would not happen. Note carefully. If A is correct, तो B is definitely correct. तो फिर तो कोई गलत नहीं होगा. It means B is correct and A is not correct. Did you understand? इसीलिए for this you have to understand the basics कि what is chromosome. DNA found over chromosome. Other than chromosome, DNA is spiraling around the histone protein. So हम DNA by taking pieces of DNA will create DNA, and after that chromosome would be produced. So first the DNA should be created, no, then chromosome. तो इसमें से पूछा है what is wrong. तो in the current level of development ये नहीं हुआ. अगर ये हुआ होता तो ये भी हो गया होता. So we would not have found anything wrong. Got it? Okay. Question number 16. What is the answer? A is the answer. Stick with English, sir. Okay, sorry. I'll stick with English. Question number 17. In the context of development of bioinformatics, the term transcriptome we had understood. Group of all the mRNAs which will definitely tell us what are all the genes which are going to get expressed, right? A range of enzymes used in genome editing, no. The full range of mRNA molecules expressed by an organism, yes, right. The description of the mechanism of gene expression, no. A mechanism of genetic mutation, no. B is the answer. Are we all comfortable with it? Transcription, do we understand? Transcriptome, do we understand? Okay. Question number 18. CRISPR Cas9 कौन बताएगा? आप बताएंगे? आपका कोई डाउट भी था ना, आपने बाहर पूछा था मैं. Okay. What is your name? Karthik ji, please answer. Consider the following statements from CRISPR Cas9. One and two. It's a genome editing tool which is simpler, faster, more versatile and cheaper than other prominent genome editing tool. Yes. The tool is inspired from mechanism used by various bacteria to fight various bacteriophage. 2 is also correct. The 2000 Nobel Prize for Physics was given to 2 scientists who developed. 2020 and chemistry. Not physics. 2020 वाला Nobel Prize था and chemistry वाला था. So this is factual छोटा सा. This is wrong. 1 and 2 are correct. A is the answer. 18th A is the answer. Okay. Question number 19. CRISPR Cas9 का ही है. Consider the following statements. You would answer it. What's your name? Vanika ji, बताइए. So far, CRISPR Cas9 hasn't shown any success in curing genetic diseases. B is the answer. You're saying 2 and 3 is the answer. Okay. 1 is wrong. We have seen CRISPR Cas9 का बहुत सारा treatment. Okay. Indian government has approved a 5-year project to develop CRISPR to cure sickle cell anemia. Yes. CRISPR has already been used for developing improved variety of agricultural crops like tomatoes. जापान वाला एग्जाम्पल, नो एग्जाम में जापान वाला एग्जाम्पल है, टोमेटोज नहीं आएगा, एग्जाम में जेनरिक आएगा, has this been used for this, or where can CRISPR be used? So these examples would help. CRISPR can be used for genetic modification. We have already seen that commercial production of tomato using CRISPR is being used, so that will give you confidence कि हाँ, this is there. Question number 20. This was another question that was asked in preliminary examination. Now you will notice कि अभी हमने बेले एक्सपेरिमेंट या ब्लॉकचेन टेक्नोलॉजी पढ़ानी है. We would study it. Blockchain will study in computer and IT, and Bele will study in physics wala portion. But CRISPR-Cas9 particle physics is wrong. CRISPR-Cas9 is about biotechnology. 3 is wrong. If 3 is wrong, 2 only. So that is why do not get panicked when you look at a question which you don't know about. That is not needed. Read the question carefully. If after reading it carefully with full patience you feel like ये नहीं कर सकते, तब छोड़ देंगे. अगर एक भी आता है, एक पोर्शन भी आता है, यूजिंग दैट ट्राई टू एलिमिनेट अदर पार्ट. ओके, नाउ क्वेश्चन नंबर 21. व्हाट इज Cas9 प्रोटीन दैट इज ऑफन मेंशन्ड इन न्यूज? अगेन प्रीलिम्स 2019 में आया था. इट्स अ मॉलिक्यूलर सीजर यूज्ड टू टारगेटेड जीन एडिटिंग. सिंपल, ये तो बहुत इजी क्वेश्चंस है. अब देखो CRISPR पे इतने इजी क्वेश्चंस 2 बार आ गए हैं. अब इफ द क्वेश्चन गेट्स आस्क्ड, इट मे गेट a bit complicated or may become application based. They may ask you ki what is base editing? Base editing was in news recently. It refers to what? Like those are CRISPR based applications which were in news. 22. Consider the following statements about T cells. T cells पढ़ा था अभी हमने. T cells are a type of white blood cells. Yes, they are formed from hematopoietic stem cells. Yes, blood stem cells. Okay, they are also called thymocytes. T cells are called thymocytes because they are thymus gland. Okay, mutations in T cell gene may lead to T cell turning against the body and ending up destroying health cells. Yes, this is the disease. So all 1, 2, 3 and 4 is the answer. Question number 23. 22 का आंसर इज डी. 22 आंसर इज डी. क्वेश्चन नंबर 23. Which of the following statements best describe the role of B cells and T cells in? अच्छा, ये तो prelims 2022. B cells T cells क्या करेंगे? पैथोजन से प्रोटेक्ट करेंगे. That is the purpose of B cells and T cells, right? White blood cells ही ये करते हैं. Easy. These are very simple questions. Again, because you understand the basic aspect. ये तो हमने लास्ट ईयर भी कई, मतलब ये वाला मैप बना के नहीं किया था, बट इन ऑन मेनी ईयर ओकेजन्स वी हैव टॉक्ड अबाउट. व्हाइल इन हेल्थ, B cells, T cells, we kept on repeating it on many occasions. What is the application of somatic cell nuclear transfer technology? Somatic cell nuclear transfer, many copies of the adult cell, cloning, production of biolarvicide, manufacturing of biodegradable, reproductive cloning of animals, reproductive cloning of animals. C. 24. Sorry? अरे व्हाइट ब्लड सेल्स के 3 टाइप थे ना, उसमें बी सेल भी एक टाइप था. दिस इज अ टाइप ऑफ व्हाइट ब्लड सेल. गो टू दैट इमेज जहां पे हमने स्टेम सेल से वेरियस टाइप्स ऑफ ब्लड सेल्स बनवाए थे. रेड ब्लड सेल्स, प्लेटलेट्स, व्हाइट ब्लड सेल्स. व्हाइट ब्लड सेल्स में टी सेल, बी सेल, ये सब थे ना. डन, अंडरस्टूड. 24 इज अंडरस्टूड. ओके, 25. मैं थक गया, आप लोग बताओ. अरे हाथ उठाना है, बताओ. सी आंसर. वाह. At present, scientists can determine the arrangement of relative positions of genes or DNA sequence on a chromosome. How does this knowledge benefit us? Scientists can determine arrangement of and relative positions of genes or DNA sequence. तो जीन सीक्वेंसिंग साइंटिस्ट कर सकते हैं, उसके क्या फायदे हैं? ओके, 1, it is possible to know pedigree of livestock. बाय, देखो जीन सीक्वेंसिंग एक बार हो गई, सीक्वेंस हमने आइडेंटिफाई कर लिया, उस सीक्वेंस से हम फिंगरप्रिंट भी देख सकते हैं. अंडरस्टैंड दिस. उस सीक्वेंस से हम फिंगरप्रिंट भी आइडेंटिफाई कर सकते हैं. उस सीक्वेंस से कौन कौन से प्रोटीन है, प्रोटीन बनने हैं, वो भी आइडेंटिफाई कर सकते हैं, राइट? तो इट इज पॉसिबल टू नो पेडिग्री ऑफ लाइफ स्टॉक. यस, इट इज पॉसिबल टू अंडरस्टैंड कॉजेस ऑफ ऑल ह्यूमन डिजीज. देखो, नॉट ऑल डिजीज आर जीन बेस्ड. सम डिजीजेस कैन बी लाइक टीबी हो गया, और लेट्स से कोल्ड हो गया, राइट? सारे कैंसर जेनेटिक नहीं है, जरूरी नहीं है, ओके। तो इट इज पॉसिबल टू अंडरस्टैंड कॉजेज ऑफ ऑल ह्यूमन डिजीजेस, नो, है ना? 2 इज रॉन्ग। अब देखो 2 रॉन्ग होता है तो 1 and 3, राइट? फिर भी थर्ड, इट इज पॉसिबल टू डेवलप डिजीज रेजिस्टेंट एनिमल ब्रीड्स, यस, इफ वी नो द जीन्स, we know which are the diseases to which this animal may be vulnerable to, we can remove or add some genes to deal with that. So knowing gene sequence is very important. If I know, like suppose my genes are sequenced, and if I know all the 20,000 plus genes which are there, then doctors would also be able to tell me that this gene 27 which is there, no, people who have this gene 27, they are highly prone to diabetes, to start taking care of your health, or get this treatment, or get a gene therapy to make you less vulnerable to the disease. ठीक है, समझ में आ रहा है? ओके, क्वेश्चन नंबर 26। अच्छा, ये तो अभी पढ़ा नहीं ना, कोविड-19, चलिए अब पढ़ेंगे। लेट्स गो टू क्या सुबह बताया था ना, हरिवंश राय बच्चन जी ने क्या कहा है, जलने से भयभीत न जो हो, आए मेरी मधुशाला। लेट्स गो टू एप्लीकेशन। भाई 9:30 तक तो पढ़ना ही है ना ऑफिशियली भी। लिसन, 5 to 7, लिसन ऑफिशियली 5 to 7, देन हाफ एन आवर ब्रेक, एंड 7:30 to 9:30, इतना तो कम से कम पढ़ेंगे। 9:30 तक तो मेरा राइट है, उसके बाद मेरी ड्यूटी है आपको पढ़ाना। फर्स्ट डे था तो क्या हुआ, नहीं दिक्कत होती भी तो मैं रुकता थोड़ी। चलिए, now we are going to talk about various applications of biotechnology, and in applications, first we are going to talk about application in agriculture sector, where we would first thing that we'll deal with is genetically modified crops or GM crops. बहुत difficult नहीं, बहुत easy है, because we understand the fundamentals. You would see that the some things which you may have found to be very difficult in the past would be very easy. तो देखो, genetic modification. Now, what are all the things that we can achieve by genetic modification? What are all the things that we can achieve by genetic modification? We have seen the example of BT cotton. A pest was attacking BT cotton. We modified, was attacking cotton, a pest was attacking cotton, was damaging cotton. We modified that cotton by adding some gene so that it become pest resistant. It became pest resistant, right? This would increase the yield. This is an example. We have created a rubber variety, rubber which was not suitable for, rubber is a tropical crop. It was not suitable for surviving in cold conditions of Northeast India. By adding some gene in it, we have make sure that it is able to survive those conditions also. हमने उसे cold resistance भी बनाया. In the same way, we can make crops which are drought resistant. Think of this, India, or India is a country, or generally in Asia and Southeast Asia, a lot of people suffer from vitamin A deficiency. Vitamin A deficiency से night blindness होता है, right? So can we modify our food crop? Like, which is the most consumed crop in India? Rice. Rice is the most consumed food product in India. Now, क्या अब तार कुछ और खा रहे? अरे रुको. So rice is the most consumed food product in India. If we can modify that rice in such a way that its vitamin A production can go high, can it solve a lot of health issues in India? तो ऐसे ही एक गोल्डन राइस बनाया गया. इंडिया ने नहीं बनाया, इंडिया में भी इंट्रोड्यूस भी नहीं हुआ है. इंटरनेशनल राइस रिसर्च इंस्टीट्यूट ने बनाया गोल्डन राइस विच हैज हायर न्यूट्रिएंट कंटेंट. तो which will have higher vitamin A. So these are various ways in which genetic modification can play a crucial role. Let us look at each of these examples. One, these are very simple, है ना. मैं यहाँ बैठे बैठे आपको सब समझा दूंगा. पहला जाओ बीटी कॉटन पे. बीटी कॉटन, कॉटन प्लांट, है ना. These insects, army worm, tobacco bud worm of Lepidopteron group, they were harming BT cotton. Scientists realized there is this bacteria, soil bacteria, कौन सा? Bacillus thuringiensis. कहीं लिखा होगा नोट्स में, Bacillus thuringiensis. That bacteria is producing a toxin that may kill those insects. So what scientists did? They extracted this gene. कौन सा gene? Cry1AC gene. It's also written here. Cry1AC gene. आपके नोट्स में लिखा Cry1AC gene. It was extracted from the soil bacteria. It was added in the cotton. तो cotton को अब हमने नाम दे दिया BT cotton because it has a gene from BT. Now this BT cotton itself is producing the toxin which is killing those pests. Life set. Pest resistant cotton we have created. Pest resistant cotton we have created. It has increased the yield of cotton in India. Before 2000, before BT cotton was introduced in India, India was dependent on cotton import. Now India is a net exporter of cotton. Because cotton production went up, a lot of farmers were growing cotton. Why? Because earlier when they were growing cotton, it used to get destroyed. The income that they were getting is much smaller. So now not only productivity is very high, but overall land area on which cotton is grown is also very high. But some recent studies have shown that some problems have also occurred. There is increased water consumption in BT cotton. Now, do you understand the meaning of, or do you understand the idea of antibiotic resistance? जब हम antibiotics को over consume करते हैं, तो eventually we develop bacteria which are resistant to that antibiotic. So we have also ended up developing some this toxin resistant pests. Pests, generally pests सारे मर जा रहे हैं, but some toxin resistant pests have also developed. For example, pink bollworm में emergence of pesticide resistant pests have occurred. Then increased use of insecticide for controlling pests like sucking pests have also happened. For other kind of pests, pesticide का use increase हो रहा है. These are some problems which are occurring. Anyways, do we all have clarity about what BT cotton is? Do we all have clarity about it? ऐसे ही there's another food crop. There's a food crop called Brinjal. Brinjal was also impacted by the same pests. जो Lepidopteron insects थे ना, which were impacting cotton. Brinjal was also impacted by this. Same thing was done. Cry1AC gene from Bacillus thuringiensis bacteria was taken out, was put in brinjal. So brinjal became resistant to this pest. Brinjal became resistant to this pest, but now brinjal plant would itself be able to produce the toxin because it has Cry1AC gene. So this increased the production of brinjal. Brinjal जो 30 to 50% loss हो जाता था because of brinjal fruit and shoot borers, this insect, now that loss got reduced and thus overall productivity went up. The crop also cleared GEAC's biosafety test in 2009. But later, Ministry of Environment, Forest and Climate Change imposed a moratorium in 2010. So GEAC is a body, Genetic Engineering Appraisal Committee, which comes under Ministry of Environment, Forest and Climate Change. It does the technical study to tell us whether this crop should be approved or not. But the final decision is with the ministry. So 2009 में GEAC ने approve कर दिया brinjal को, but because of a lot of protest, because of a lot of opposition, Ministry of Environment, Forest and Climate Change did not allow its release. So BT brinjal, देखो इंडिया में there is only one genetically modified crop which is allowed to be cultivated commercially, that is BT cotton. BT brinjal develop हो चुका है, इंडिया ने डेवलप नहीं किया. BT जैसे BT brinjal was developed by Monsanto, the same company, the same seed company has developed BT brinjal also, but India has not approved its commercial cultivation. But still, there are examples of illegal BT brinjal cultivation going on in the country. Various studies found that in Haryana in 2019, BT brinjal seeds were being sold and they were being cultivated. Illegally, definitely वो illegally होता है. तो some groups are calling for allowing BT brinjal's cultivation or allowing the introduction of BT brinjal in India. One reason that they are giving is by GEAC approval was given to it. The expert body did its study and found that it is not problematic. Why are you creating problems? Second, increased benefit for farmers because productivity will go up. It would benefit farmers. Examples from Bangladesh have shown that productivity has increased by 40 to 50%. Finally, they also say that when officially approved BT brinjal is not available, what is happening? Illegal BT brinjals are being made available in the market. Now those illegal BT brinjals are not even going through the proper safety approval mechanism and thus may be more problematic, more dangerous. Something like if you ban alcohol, they will drink hooch. Hooch tragedy would occur. Illegally manufactured alcohol can be much more problematic. So that is going on in the country and thus we should allow the approved BT brinjal to continue. बात समझ में आई है? Okay. But BT brinjal is not being allowed because it there are many various reasons. One, we say that we have many varieties of brinjal. All of them would be replaced by BT brinjal. That would be negative. That would be negative for biodiversity. More importantly, they say that because of cross pollination, this BT feature may go to non-GM crops also. Genetically modified crops जो नहीं है, उसमें भी ये features जा सकते हैं, which is something that is not accepted. Then health impact is also something that is being studied. Like जो लोग oppose करते हैं वो कोई भी बहाना देते हैं कि अभी health impact पे study इतना सारा नहीं हुआ है. Finally, some of the studies have also shown that in Bangladesh, which had allowed BT brinjal to be produced commercially, there also some of the farmers may have gone back to the previous kind of brinjal because the output was not that great in long term. ये कुछ problems रही है, but ये थोड़ा analysis mains के लिए, pre के लिए BT brinjal, BT cotton, Cry1 AC gene from Bacillus thuringiensis introduced here, brought here. Questions related to this have been asked 3 times since 2009. सिर्फ BT वाले इशू पे 3 बार सवाल आ चुके हैं since 2009. 2009 में BT brinjal वाला बवाल हुआ. Since then, this question has been, abhi very recently in 2018 they had asked a question on this. UPSC had asked a question on this. Okay, let's go to genetically modified mustard, GM mustard. Recently GEAC has approved it. It was in news October 2022 में GEAC had approved it. So the mustard that we are going to talk about is Dhara mustard hybrid 11 DMH11. Let's talk about this. Dhara mustard hybrid 11. This mustard variety is the first food crop, genetically modified food crop which has been developed in India. DMH11 is a genetically modified food crop which has been developed in India by Indians. So there is a former Vice Chancellor of Delhi University, Deepak Pental. Who has developed this Dhara mustard hybrid 11. Now your question may be, sir, ये GM है या hybrid है? Dhara mustard hybrid, and we are studying it under genetically modified crops. What are you saying? GM है कि hybrid है? दोनों. To make hybridization possible, genetic modification was done. Now, what we actually wanted to do was this. We wanted to cross the Indian mustard with the European variety of mustard. So what we wanted to do was this. There is this Indian mustard variety. We wanted it to be crossed with European mustard variety. Cross करके दोनों से acceptable features would have led to production of better quality of seeds, better quality of mustards. That is what was what we wanted to do. But the problem that occurred was this. This is self-pollinating in nature. Self-pollinating in nature means खुद को pollinate कर लेता है. So now how do hybridization take place if it gets self-pollinated? How does hybridization take place if it gets self-pollinated? So first what we had to do, we had to stop its self-pollinating property. So a gene was added to it. This plant was genetically modified to introduce Barnes gene. आप देखेंगे यहाँ पे 3 जीन्स इंट्रोड्यूस करे हमने, बार्नेस, बार्स्टर एंड एक और जीन है बार जीन. तो बार्नेस जीन was first introduced in which variety? In the Indian variety. To do what? To stop its self-pollinating property, to make it male sterile. Now it will not be able to pollinate itself. It became male sterile. So इसका self-pollinating property was stopped. अब ये self-pollinate, now this is not self-pollinating, so it can be pollinated by some other species pollen, and thus hybridization is possible. But अगर हम ऐसे ही hybridization करा दें, the species that would be created will also have Barnes gene and will be not self-pollinating. But we want that final species should be self-pollinating, the way mustard is. And we want to create a hybrid, but the final species should be self-pollinating. So what we did, हमने बोला ये जो है ना यूरोपियन, इसमें add a gene called Barster gene. ये बार्स्टर क्या करता है, ये बार्नेस के फंक्शन को रोकता है. बार्नेस जीन था ना, बार्नेस, ये बार्नेस के फंक्शन को रोक देता है. बार्स्टर जीन इसमें डाला अलग है, इसमें बार्नेस डाला अलग है. इट इज नॉट सेल्फ पॉलिनेटिंग, so it can be cross pollinated with this version. एक हाइब्रिड बना. When hybrid got created, it has Barnes gene which does what? Which makes it male sterile, which makes it कि भाई pollination नहीं हो, self pollination नहीं होगा. But then Barster comes with the European variety. It says Barnes काम नहीं करेगा, Barnes will not work. Barster will stop the functioning of Barnes. तो Barnes का function stop हुआ, मतलब it becomes self pollinating. So hybrid became self pollinating. Hybrid became self pollinating. So we have created a hybrid, but it has 2 genes, foreign genes. Genes और भी हैं, Barster gene and Barnes gene, which allowed cross pollination, but ensured that in the end we have a self pollinating mustard, a self pollinating mustard variety. Got it? सबको समझ में आया? Anyone who hasn't understood it? नहीं? पक्का? Urvashi, did you understand? पक्का? Will you be able to repeat it? No. Okay, I'll repeat it for you. Pen do. 2 mustard varieties, the Indian variety, the European variety. We wanted to create such hybrid, but this Indian variety is self-pollinating. So इसका hybridization with the European variety was not possible because it used to pollinate itself. So we added a gene in it called Barnes gene, which makes it male sterile, which stops its pollen production and thus makes it not self-pollinating. It cannot pollinate itself now, right? Then we hybridize it. But when we hybridize it like this, यहाँ पे Barnes gene है, it will lead to this product not this hybrid not remaining self-pollinating. We did not want that. So before hybridization, it has Barnes, which makes it male sterile. In this we added Barster. Barster does what? It stops the functioning of Barnes. तो अभी तो Barnes अलग है, इसमें Barster नहीं है, so it is not self-pollinating. We pollinated them. It has Barster. We pollinated them. We created a crossbred. Crossbred has been created. Barnes was there, but Barster is also there. So Barster will stop the functioning of Barnes. What happens? It becomes self-pollinating again. So we have created a hybrid variety which has good features of both and is self-pollinating as well. अभी समझ में आया सबको? पक्का? DMH mustard. I promised you. आप लोग जाके ना जिन्होंने मेरे से पढ़ाई नहीं करी है उनसे पूछना धारा मस्टर्ड हाइब्रिड 11 कैसे बनाए, आधे लोग नहीं बता पाएंगे, बिकॉज ये अच्छे से बहुत बेसिक चीज है, बहुत छोटा सा चीज है, whether it's hybrid or GM, how this hybridization was achieved, but people do not study it properly and therefore they end up facing problems. I remember in 2018 I had just started teaching. मैं जिस पुराने इंस्टीट्यूट में पढ़ाता था वहां पढ़ा रहा था, एंड वाइल टीचिंग आई सेड कि ये इंडिया में बना है, दिल्ली यूनिवर्सिटी में बना है, दिस काइंड ऑफ थिंग्स, एंड देन उस दिल्ली यूनिवर्सिटी के रिसर्च लैब का नाम भी बताया था, उसमें बना है, एंड देयर वर पीपल, बिकॉज उस टाइम ना मेरा वेट थोड़ा कम था, तो आई यूज्ड टू लुक यंगर, देयर वर पीपल हु वर जजिंग मी, दे वर लाइक अरे इसे पता नहीं क्या क्या पढ़ाना है, कुछ भी पढ़ा रहा है. दैट ईयर इन द एग्जाम दे एग्जैक्टली आस्क्ड, ये एक स्टेटमेंट जो दिया था वो ये था, ये पंजाब यूनिवर्सिटी में बना है, which was a wrong statement because it was made in Delhi University, but UPSC had asked those things. So UPSC has already asked questions on DMH 11 or Dhara Mustard Hybrid 11, and they may go into more details. They may check your conceptual clarity on whether you have understood this genetic modification has been done or not, and for that reason you have to know it very clearly. Superficially नहीं पढ़ना है किसी भी चीज. You have to clearly understand what is happening. Fine, Dhara mustard hybrid 11 is done. Then there are some more genes which are added for marker and all, not very important. So this method was used to develop DMH 11 by crossing popular Indian mustard variety Varuna with the East European variety early Hera 2 mutant. So Varuna and early Hera 2 mutant were merged together to create Dhara mustard hybrid 11. Got it? Now, जो ये बार्नेस एंड बार्स्टर जीन आए हैं, they have not come from BT cotton or BT or Bacillus thuringiensis. They have come from bacteria called Bacillus amyloliquefaciens. It's a different bacteria जिससे हमने क्या लिया, बार्स्टर एंड बार्नेस जीन लिया. So that's why it's not BT mustard. Why not BT mustard? Because वो Bacillus thuringiensis से नहीं आ रहे हैं. They've given a different name to it, Dhara mustard hybrid 11. The bacteria from which the gene has been taken is Bacillus amyloliquefaciens. Fine? Clarity? Okay. Now, argument against approval of GM. Argument for and against approval. The for, why it should be approved. Higher production. देखो, India is a country, though for we are a net agricultural product exporter, but when it comes to edible oil, we are dependent on import. A lot of edible oil gets imported in India. Now, mustard is something that is primarily used for making edible oil. At least in North India, like हमारे घरों में आज भी सरसों तेल में खाना बनता है. सरसों तेल is this common. Sarso is mustard. Mustard oil is this common. Nowadays, we have started using that refined and all those things, but traditionally in rural areas, it is still mustard oil that is consumed. It is the most consumed edible oil product. Traditionally, it is the most consumed edible oil product. So if we can increase the production of mustard oil by 30-40%, it can be beneficial for us. So higher production of mustard will lead to reduced import dependency, will also save foreign exchange. When we are importing things, we are spending our foreign exchange, keeping India scientifically relevant. Why? This is important because if we do not approve our genetically modified crops, see, look at Japan. CRISPR अभी डेवलप हुआ, टमाटर बना के उगा भी रहे हैं, खा भी रहे हैं और एक्सपोर्ट भी कर रहे हैं. एंड डू यू नो, इंडिया इज ऑलरेडी इंपोर्टिंग अ लॉट ऑफ एडिबल ऑयल which is genetically modified. We are not growing that edible oil. We are not growing mustard which is genetically modified, but we are importing edible oil which is genetically modified with the same gene. बार्नेस बार्स्टर जीन से जिन ऑयल में है ना, हम वो कंज्यूम कर रहे हैं. वी आर इंपोर्टिंग इट. सी हाउ सिली दिस इज. एंड इफ वी डू नॉट अप्रूव इट, व्हाट वुड हैपन? साइंटिस्ट्स हु आर वर्किंग ऑन दीज थिंग्स, दे वुड फील बैड, दे वुड सफर, दे वुड गेट डिस्करेज्ड, दे वुड बी लाइक छोड़ो इंडिया, यूएसए चलते हैं, ब्रेन ड्रेन, दीज प्रॉब्लम्स. अगेंस्ट वही है कि बार्नेस बार्स्टर सब illogical arguments. Leave it. GEAC has recently approved it. GEAC has not given approval for commercial production but for environmental introduction. once GAC gives approval for environmental introduction in a controlled way, it could be grown and then उससे seeds collect किए जा सकते हैं and then later commercial production may happen, but Supreme Court has put a stay on this. So you'll go through this, read it, understand the reasons behind all the conditions that have been put here. GAC ने जो approval दिया है environmental introduction का, उसमें a number of conditions have been put. So why those conditions have been put, those kind of things, please understand. From science perspective, I hope all of us have clarity about what is Dhara Mustard Hybrid 11. GM rubber, genetically modified rubber. What is GM rubber? तो आप देखेंगे Rubber Research Institute has developed a plant tailored for climatic condition of Northeast. Rubber was a tropical crop. Rubber Board Research Farm at Sarutari on the outskirts of Guwahati now sports world's first GM rubber plant tailored for climatic condition in Northeast India. What was the genetic modification done? The GM rubber has additional copies of gene MNSOD or manganese containing super oxide dismutase. This is the gene जिसकी additional copies हैं किसमें? GM rubber which we have created, inserted in the plant, which is expected to tide over the severe and cold condition during winters of Northeast India. This is genetically modified rubber. Now you understand how gene may have been introduced and all. उसमें ज़्यादा टेंशन लेने की जरूरत नहीं है. Similarly, एक एग्जांपल है न्यूट्रिएंट कंटेंट इंप्रूव करने का, गोल्डन राइस. तो इन गोल्डन राइस as we said कि विटामिन A deficiency causes what? Night blindness. And India, Southeast Asia, these are the places which suffer heavily from vitamin A deficiency. Generally, poor countries or countries which have a lot of people who are not able to afford fruits and all, they suffer from these diseases. Vitamin A के लिए आपको fruits वगैरह, green vegetables वगैरह consume करने पड़ते हैं. So when there is poverty, people are not able to consume. They suffer from it, and therefore, International Rice Research Institute. Please note that International Rice Research Institute इंडिया में नहीं है, इंडिया ने नहीं बनाया है. India is not growing golden rice, but they have developed what is called golden rice. It looks something like this. Golden in color, जैसे orange वगैरह rich in vitamin A, है ना? तो somehow like that. So what does it do? Golden rice is a variety of rice produced through genetic engineering to biosynthesize beta carotene. It produces more beta carotene. This beta carotene is a nutrient which eventually will become vitamin A as and when needed by body. This beta carotene is something that is found in orange colored fruits and vegetables. ये विटामिन ए इससे बनेगा. तो गोल्डन राइस इज दिस राइस. तो अगेन an example of genetically modified crops which has led to increase in nutrients. पल्लवी को भी नींद आ रही है, सो जा. यहाँ पे we have given some detail about International Rice Research Institute. ये भी ना कभी कभी प्रीलिम्स में कुछ भी पूछ लेते हैं, तो you should know about who has developed it, where is it located, and India has not started producing it, but Bangladesh and some other countries have started doing it on an experimental basis. Now illegal cultivation of GM crops वाला issue, हमने बीटी ब्रिंजल की बात करी थी कि बीटी ब्रिंजल का illegal cultivation हो रहा है. Then जून 2021 में एक और controversy हुआ था. In Europe, the rice which was exported from India was found to contain some amount of genetically modified rice. India said that it may not have happened from India. आप जहाँ पे उसे process कर रहे हो, crush कर रहे हो, some other rice from Europe and all may have been included there, which has led to our product having genetically modified content. Our product having genetically modified content. But India had formed a committee here to deal with it. कि क्या was the rice from India genetically modified rice, or is India growing, or are Indian farmers, some of Indian farmers are growing genetically modified rice in an illegal manner. ऐसी एक और issue है herbicide tolerant BT cotton वाला. तो देखो BT cotton में what is there? Bacillus thuringiensis bacteria. Oh sorry, Cry1AC gene from Bacillus thuringiensis bacteria. It is there. Now this bt cotton can protect itself from those pests, right? Lepidopteron वाले group के pest से it can protect itself. Now Monsanto has developed another variety of bt cotton called herbicide tolerant bt cotton. Herbicide tolerant, HT, herbicide tolerant bt cotton. Now what is herbicide? To understand herbicide, do we understand what weed is? Not वो फूकने वाला weed नहीं. Weed जैसे agricultural crops के साथ जो unwanted vegetation that grows, that weed. Okay. Now those weeds could be killed by herbicides. Herbicides are chemicals which can kill those weeds, right? But when we put herbicides in our field, they also kill or damage cotton crop. So to use strong weedicides or herbicides which can kill those weeds, we first want to make our cotton plant tolerant to herbicide. We first want to make our cotton plant tolerant to herbicide. Herbicide tolerant bt cotton. Did you get why herbicide tolerant bt cotton would be needed? Do we have clarity on this? Herbicide सब एकदम मरे मरे से हो गए हो हैं? ये देखो, ये गद्दा मंगवा देता हूँ, रिक्लाइनर ऐसे आराम से, है ना, ऐसे मैं मुजरा का टाइप भी कुछ कर दूंगा, तो वैसे नाचो मेरी जान करना मेरे को देख के। और लग जाओ, ऐसे थका हुआ लग रहा है, मर गया है, नींद आ रही है। बीस मिनट पहले क्यों आ गए थे? क्या करोगे घर जाके? इतनी सर्दी है, किसी का पढ़ने का मन तो करता नहीं, सीधा रजाई में घुस के सो जाते हैं। एक अकेला मैं ही हूं जो पढ़ रहा होता हूँ, नोट्स बना रहे हैं मुझे कल के। क्या करोगे घर जाके? भगा दूंगा मैं, ऐसे फालतू की बकवास करोगे तो। चलो, डिड यू गेट एचटीबीटी कॉटन वाली बातें? एचटीबीटी कॉटन में क्लैरिटी है। तो सपोर्टर्स हु आर सेइंग कि एचटीबीटी कॉटन यूज करना चाहिए, दे से दैट इट विल सेव कॉस्ट। व्हाई विल इट सेव कॉस्ट? राइट नाउ टू रिमूव वीड्स व्हेन यू यूज लेबरर्स का कॉस्ट बहुत ज्यादा पड़ जाता है, एंड जस्ट इट इंक्रीजेस द कॉस्ट ऑफ प्रोडक्शन ऑफ कॉटन। तो फार्मर्स ग्रुप्स लाइक शेतकारी संगठन आर डिमांडिंग लीगलाइजेशन ऑफ एचटीबीटी कॉटन। बट एक और रीजन है व्हाई इट इज सेड कि लीगलाइज करो, बिकॉज इलीगली अवेलेबल एचडीबीटी कॉटन आर ऑलरेडी बीइंग यूज्ड। नाउ जो इलीगली अवेलेबल है उस पे सेफ्टी मैकेनिज्म एंड ऑल दोस थिंग्स मे नॉट बी फॉलोड, इट मे बिकम अ कॉज ऑफ प्रॉब्लम। बट देन देयर आर ऑपोजिशन आल्सो। Glyphosate have the weedicide that we are using here is glyphosate. Now glyphosate has recently been banned, so it was the weedicide जिससे हम tolerant बनाते, but this weedicide as such will have a lot of negative environmental impact. This was the people who were opposing HTBT were saying you should not use weedicides or you should not use herbicides. They are damaging. Manual mechanism is better. Unchecked spread of herbicide resistant may create problems for more crops, and weeds may develop which are resistant to herbicides. It may become bigger problem. Okay, these were genetically modified food crops. Okay, तो जो मेरा राइट था वो खत्म हो गया, मैं ड्यूटी फुलफिल कर लूं? अच्छा, कितने लोगों को नहीं पढ़ना है? अरे नहीं, अरे मैं डांटूंगा नहीं, उठा लो। ओ, मैंने ऐसे देखा। कितने लोगों को और पढ़ना है? दो उठा रहे, तू तो झूठा है। हाँ, उठाओ, कितने लोगों को पढ़ना है? और नहीं पढ़ने वाले कितने? तो बाकी क्या है जो बीच वाले रह गए हैं? नहीं, ब्रेक नहीं, एक काम करते हैं, रुकते हैं, विल स्टिक टू दिस। हम एक्स्ट्रा क्लासेस लेंगे, रादर देन सैटरडे संडे या फ्राइडे सैटरडे संडे। एक दिन में 4 ही घंटे पढ़ते हैं, वरना पागल हो जाओगे। ठीक है, तो आई विल सी यू टुमारो 5 PM प्लस प्लीज इंश्योर 5 बजे ना आप लोग सेटल्ड हो क्लास में, सो दैट आई कैन स्टार्ट द क्लास। हाँ हाँ, मैं बोलता हूँ, मैं सर से बोलता हूँ, तो 5 बजे आई वांट द क्लास टू स्टार्ट। ठीक है, और द स्टूडेंट्स हु आर वाचिंग इट ऑनलाइन, प्लीज टुडे इज द लास्ट डे फॉर वो 23% डिस्काउंट वाला, तो प्लीज टेक एडमिशन टुडे ओनली इफ यू वांट टू टेक एडमिशन। थैंक यू वेरी मच।